All right. All right. Welcome back, everyone, online and in person. We're going to continue on to our data analysis workshop. This is, sorry, we're running a little bit behind, but things are, as you know, can be kind of crazy in person as we're trying to get used to doing in-person things again. So I hope that everyone that's watching remotely had a little time to get some lunch or dinner or breakfast, depends on what your time zone is, I suppose. All right, so our next speaker discussing high-dimensional data analysis workflow is Nicolas Loof. He's returning to discuss this unsupervised analysis using algorithms in FlowJo version 10. I obviously know Nick going back quite far. He's been a part of the FlowJo or FlowTex committee and community for quite some time. He has over 10 years experience in the field of flow. He's currently a senior application scientist, multi-omics specialist at BD Biosciences. Previously, he served as an SRL manager at the Baylor Institute for Immunology in Dallas. And in 2012, he joined UT Southwestern in Dallas as the director of the Moody Foundation flow cytometry facility, where they serviced over 120 different laboratories. In 2021, Nico then went to the dark side and joined FlowJo, which was then also picked up by BD, and is currently, he's a recognized member of ISAC since 2010. He's a former member of ABRF, but more importantly, he's one of our committee members at FlowTex, which makes him awesome. So without further ado, Nico.
Thank Joe for the introduction. All right, so you have my email address here. If you have any questions, don't hesitate to reach out to me. I have no problem. I try to answer the email as soon as I can. So it's going to be a brief overview of the software, how to analyze the data. I'm sure you're going to have plenty of questions after the talk. And if you was not able to answer any of them, don't hesitate to email me. All right, so for me, data analysis is a little bit like a chess game. All right, so you need to move all the parts in a different way to be able to win your game at the end. And first, you're gonna have to move the pawn, and after you're gonna have to move the queen and everything to try to win your game. So let's go into different detail. So when I start flow cytometry in the lab, we were using biaxial plot or 2D plot to analyze your data. Thing is, now with the high parameter flow cytometer that we have, we can do 30, 40 parameter flow cytometry and just do the dot plot, two-dimensional dot plot is not sustainable anymore to analyze our data. We have so many end-by-end plot available that if we spend 2 minutes per plot for 40 color, you're gonna spend at least 17 hours to analyze the data, and it's just not possible. So we try to move from dot plot to the end parameter hyperspace using TCD, for example, but any other algorithm to do dimensional reduction and clustering algorithm. But sometimes, you make a good discovery using those tools, and you're like, well, how get back to be able to solve that population using 2D plot? And there's this tool now that's been developed to be able to go back and forth between 2D plot to hyperspace analysis. So all the tools I'm gonna talk about today are available for free on our FlowJo Exchange website. So just go on our website, flowjo.com/exchange or exchange.flowjo.com. Both of them work. You can download them, install them into FlowJo, and you're gonna be able to use them. Those plugins are either created by third-party person or developed inside in our team at FlowJo. Some are better than others, and some are being updated. Some are not, because if it's not developed by us, they might not be updated. All right, so here's the workflow to analyze data. for high parameter analysis. So first you're gonna do data QC, then we're gonna do normalization, annotation, concatenation, dimensional reduction, clustering, phenotyping, comparison. And if you want to sort, you can do at the end isolation. So the first four, I really go hand to hand together, is really just to make sure that whatever cells you put in your analysis or whatever event you put in your analysis, they're good quality, because if you put garbage in, you're gonna get garbage out, doesn't matter the algorithm you're using. Dimensional reduction clustering, they're not really as useful by themselves, but together that's really where the power comes from. Phenotyping comparison, that's you guys as the scientists that know what you're looking for that come into play, and there's no really any algorithm that can help you with that. All right, so first things, we need to clean up our sample, make sure our sample are clean. So we're gonna move our pound to clean up our sample. So our first algorithm we can use, it's the cleanup algorithm. And there's 3 available right now at FlowJo: FlowAI, Peacock, and FlowClean. They're used to remove any unwanted events, so clog, air bubble, anything that happen on the instrument. So depending on which algorithm you use, they look at the dynamic range, the flow rate change, and the signal acquisition change. So your flow rate, if you run at 5,000 events a second, doesn't matter for how long your event rate should be stable. It should not drop or get up. But the same thing for the signal acquisition, your MFI, because your cells coming randomly into the instrument, your MFI should be stable over the time. I get a lot of questions saying which one is the best between those 3 algorithms. And I think that's everybody's question in mind. My personal preference is FlowAI. Doesn't mean it's the best. That's the one I use the most and can rely on, in my opinion. FlowClean is the oldest version, is the oldest algorithm to clean up your data. The way it does it, it just separates your sample in a really small time bin and split the sample in 2, positive and negative. Doesn't even matter if it's true or not, and making sure that the percentage between each time bin doesn't change. FlowAI, on the other hand, is looking at all 3: dynamic range, flow rate, and signal acquisition. Might be really strict, but I think I like it. Peacock is the last one, been published, if I remember correctly, 2021. Get that wrong, maybe, I'm sorry. So newest technique, in my personal experience, I'm not confident enough to use it regularly on the sample, but I'm sure it's a decent or good algorithm, knowing from the lab is coming from. All right, so now our sample is clean. We want to do composition. Same thing, composition. It's a big deal, a lot of questions about composition. You have many ways to do composition in FlowJo. You have, of course, the traditional composition, the auto-spill composition. You have spectral, so traditional auto-spill can be spectral. You can remove autofluorescence with auto-spill. And you can compare the different composition to know which one is the correct one. You can also look at the spread matrix, as I mentioned this morning, to make sure that your panel is the best panel that you can design. Because if you want to do 40 colors experiment, you need to make sure that you can distinguish all the different population and don't have too much spread. Now we're going to the heart of the analysis, so normalization. So when you run samples, you're usually not going to run all your sample on the same day. You're going to run sample on different day, depending on when the sample are coming from the patient or just when it's time available on the machine. So when you run sample on different day or on different instrument, what can happen is you have batch effect. So if you have batch effect, you need to remove that batch effect to not having the analysis affected by that batch effect. So you have Cyctonome, which is an algorithm to remove the batch effect. It's going to make sure that the control that you run between each instrument or each different day that you mentioned here with the cross, just to make sure this sample is exactly the same between each run. If it's not, it's just going to adjust the sample to make sure it's correct. And you can see on the CD25 signal right here, that was, sorry, CD66 signal, the batch one was a little bit lower. Signal, if I use the laser, it will be easier. CD66, batch one, the MFI is lower. After normalization, the MFI is the same. In my opinion, don't normalize the data if you don't see batch effect. So we saw that for few users when we help analyzing the data, that normalizing the data when you don't have batch effect can minimize your biological differences between samples. So analyze the data with raw data. If you see batch effect, and it's really easy to see when you have batch effect, just come back and do the normalization. All right, annotation. I cannot emphasize this slide enough. Just annotate your data. I'm tired to see sample 1, tube 1. I don't know what it is. And especially if there's no keyword, nobody knows what it is. So make sure to label your tube, make sure to annotate, use keyword. Use keyword on the instrument. That's where your keyword is going to be integrated into your FCS file. So you can change your FCS file and everybody's going to know what is it exactly. If you haven't done it on the instrument, do it on any acquisitions, so, any analysis software like FlowJo. We have way to create keyword, add your keyword. Even using the plate editor, you can just put your Excel file in it, and all your keyword would automatically be added to your sample. The difference by adding your keyword in FlowJo compared to on the instrument is in FlowJo, we don't modify your FCS file. So you're still going to have your raw data without the annotation. Or if you want to share your data, you're going to have to share the FlowJo file for that person to see all the annotation. All right, so now that our samples are clean, normalize, if you have to annotate, we can concatenate our sample. And concatenating sample is because of the nature of the algorithm. The nature of the algorithm, the way they seed at the beginning, your cells to put them on different spots, or when you do clustering or visualization, they have to be run all at the same time. And so we're going to concatenate all the samples into one sample, one huge sample. We're going to annotate each one of the samples that we've done to make sure that we can separate our sample again in the future after the analysis, after running the algorithm. And we're going to have this huge one file that we're going to run all the analysis. All right, so sample is QC, we're good to go. Now let's move our king, rook, do the bishop rook, advance, move the queen to try to see what's going on. So first one is dimensional reduction. And dimensional reduction is just to visualize your data. You cannot look at 400 and something dot plot at the same times, but you can look at a two-dimensional X, Y, Z plot with the different cell type represented here. Each island represents a cell type, or at least a group of cells that are similar to each other. So here there's a group of cells, here they're manually overlaid. So we have the CD66 right here on Disney, UMAP, TriMap, BackMap. There's many different dimensional reduction tools. They have all pros and cons. Disney, really good to look at small population, but the distance between each island has no meaning. If you look at UMAP, the distance between each island has a meaning. Two islands that are really close to each other, that means those cells are really similar to each other. Same thing for TriMap, BackMap, pros and cons there. So look more into the paper if you want to know more. Not gonna go too much in detail today between those differences. So also when you do your visualization, you may want to be able to automatically, by the visualization, tell which cell type it is. So you can do a heat map on the expression. So that's what it is here. So we use the Disney, and we do color-coded expression of each marker. So here for IL-1 beta, we see that the island here on the left is IL-1 beta positive, that means that's my L1 beta cells and everything else is negative. And I can do that for each marker. I can see my CD14 positive here on top is the big island or the big potatoes, way you want to describe it, scientific or not, there. Okay. So that's a good visualization. That's not giving you really good analysis. It's just for visualization when you go to presentation like today and you want to say, oh, it's my one beta that I'm going to talk today, and here they are. The way, instead of getting around each island manually, the way to define those populations is to run the clustering algorithm. That's what I meant earlier. That's really go end to end. So the clustering algorithm really gonna draw a line around each cluster, and you're gonna be able to define those populations. And of course, it's way faster than just drawing those gates manually. And it also may offer a new perspective. So maybe you think it's only one big potatoes, but in fact is two they're really close to each other. Why I'm presenting FlowSum first here. FlowSum is the most used currently in the field. Same thing doesn't mean it's the best. That's just currently the most used. He gets, I think, one of the main reasons is used the most is because he has visualization. So you get this spanning tree result that really can tell you the different cluster and what the expression of marker. The two other algorithms that you can use is phenograph and X-shift. Phenograph is the Swiss knife, not great to anything, but is good, is average to everything. So if you don't know what you're looking for, I think start with phenograph, and after, if you want to do really analyze on the specific subject, maybe switch to either FlowSum or X-shift. X-shift is really good at finding rare population. So if that's your research that you're doing, X-shift is the way to go. All right, so now we define all the cluster by the algorithm. I'm gonna be able to define it as a scientific point of view. So, of course, you can look at the expression of each marker individually, but we find easier to create another algorithm, which is called Cluster Explorer. And Cluster Explorer is just gonna give you the profile. Each line is a profile of cells and giving you the expression of each marker. And you're gonna be able to gate on each of those markers. So I can select my CD4 positive cells, CD3 positive cells, and it's gonna give me all the cluster that meet those criteria. And as a scientist, I can tell these cells are my T helper or whatever cells you want to define. It's going to also give you a heat map if you'd rather to have heat map. And also you're going to have a visualization on the dimensional reduction. So you can even click on the island to see which one is highlighting to exactly define, and maybe even compare the different clustering algorithm, because on your dimensional reduction, those spots never move. So if you click one, you can look at FlowSum, X-shift, phenograph, and compare if cluster 1 from X-shift is maybe cluster 3 for phenograph, etc. All right, so now we did the analysis. We want to compare between all the different experimental conditions. So same thing, you can do it visually. So you visualize your data, you just split based on the different condition and see maybe where there's differences. Okay, here I have things that disappear in my severe patients, and it's all there not stimulated, and it's back in my convalescent patient, etc., etc. That's one way to do it. I think that's maybe not the most efficient way, but that's one way that can be easy when you want to do visualization. You can also see differences between the different stimulation. So here, difference between no stimulation, stimulation. And now we go to the Queen advance to kill the king, and we really want to go on Cluster Explorer to see the differences. We can import or gate between the different conditions and see exactly what's overexpressed, underexpressed, or what didn't change between all different conditions. You have other visualization available. One of them is variant blocks that give you a really nice graph there. And at the end, you probably want to isolate the cell population that you define, and you're gonna use the HyperFunder plugin to isolate this population. And you're gonna define that cluster. You say, I want to define this cluster. You're gonna tell in how many gates you want to define that cluster maximum. That's just because some software or the sorting software, they're limited in a certain number of gates. If you have a fax area, for example, you're limited to 8 gates to define that population. So you can define this cluster in at least 8 gates, but maximum 8 gates to be able to sort that population. And because BD and FlowJo are now one, when you define that population using the Cluster Explorer, you can just export this getting strategy directly to FaxDiva to be able to sort that population. Yeah. All right, the references of what I'm talking, and just want to make sure when usually I give that talk, people are worried about their job. Your job's not going away. It's not because you're not going to spend 17 hours analyzing the data that your job is going away. We can do data sorting, arrange, present visually. That's what software can do, but we can never explain the story without you. All right, so let's go to the demo, the software. Session open. All right, so open FlowJo. You go in your workspace tab, you have your plugin tab, and you see all the plugin I've downloaded on mine. If you have any issue or question about installing plugin, just email me or contact tech support at flowjo@bd.com, and we will help you how to install the plugin. So the data we're gonna use today are the OMIP60. Here's the paper. OMIP60 is a 40 color data on human PBMCs. And what they did is they did manual gates to define all the population. So we have our basophil, our T cells, B cells, dendritic cells, everything. And also they did the hyperparameter analysis. I don't want to know how much time they spend on doing the manual gates. Maybe Diana can tell us, but I will hate that than that. So let's do the hyperparameter analysis and let's compare how it is compared to their manual gating to see if we can define the same population. Or maybe we find something new that they didn't see during the manual gates. All right, so opening the data, you can drag the file into FlowJo. I'm gonna so it's SciTech Aurora data. So they're already mixed, so I don't have to do the composition. So I'm just gonna put everything that's not sample in my composition folder. I'm gonna just create a new group for my sample. And they call MC in that sample. So this one is test. I can organize my sample. I'm just gonna use, just gonna create a group and click and drag those into the sample. Easier. Just 4 samples. All right, so I have more 4 samples here. So first step I want to do is to clean up my sample. Okay. So when you look at the paper, they just did a manual cleanup. So they just did a time gate, which we used to do that in the past, but now with the algorithm, it's better, it's faster. use the algorithm. So I'm just gonna use FlowAI, as I mentioned. That's my favorite one. There's nothing wrong using any other one, but I'm gonna use that. I like it. Ask you to save your workspace. Yes, I'm gonna save it. But it's something I can remember. Delete later. On my desktop, FlowTex. And I'm gonna ask me which parameter I want to use to remove the clog. So I'm gonna use every parameter uncompensated. So there's a big difference. Peacock requires compensated parameter compared to FlowAI, and FlowClean doesn't require one raw data. So yeah, I'm gonna use the raw data, and I can choose which anomaly to exclude. Do I want to exclude flow rate? Do I want to exclude signal acquisition? Do I want to exclude dynamic range to make sure that I'm within the linear range of the instrument? Or just I want a combination of two or all of them? I'm just going to select all of them. Numbers, you can change them. I don't recommend it. They're set for a reason. Just leave it there at the beginning. If you want to play later on, when you put your mouse over it, it's going to tell you exactly what happened and how to change that. I'm going to remove the outliners, and I'm just going to run the algorithm. So it's opening my FlowAI node here, calculating. It will take about 30 seconds to 1 minute to run for that sample. If my computer wants to cooperate, because we have Zoom running as well. It may take a lot of memories. Anybody has a music? No. Go ahead. So I run all check because I want to remove everything. If you realize that you remove way too much, maybe because you're on an instrument that you can change the flow rate when you're running the instrument, but you're gonna remove the flow rate check, for example, maybe on one specific sample, on all your sample, depending if you change it. If you run site of data, you don't have flow rate on site of data, don't select the flow rate for site of data. But if you select only dynamic range and the signal acquisition, you can run site of data on FlowAI. So it really depends on what you run and what you see. You're welcome. All right, so we run FlowAI. So we have the node, we have the derived parameter that we created. I'm gonna have to talk like that for the microphone. And you have the good and bad event. So for the good event, it kept 63% of my sample, and it removed 36% of my sample. Maybe looks a lot for most of you, right? Why you remove almost 40% of my sample? Well, we can check. So let's go in the folder that I just created with the name, have my FlowAI, and I can go in the FlowAI here. You can go in the figure, or you can open the HTML. Usually I like to open the HTML better. So here's the picture. If you want the picture, I like to open the HTML. I think a browser, it's easier to navigate through than just the picture. All right, so it give me, for my sample, this sample, it removed 36%. I have the name of the sample, number total of event, and it removed 1.61% due to the flow rate. So really at the beginning, kind of makes sense, you know, your flow rate cranked up. And after it's stable, there's some fluctuation that it removes really specific point, maybe a small clog or burst of cells came, so it moved less than 2% in flow rate. With that, nothing to complain. But it removed 32% in my signal acquisition check. Okay? So that means my signal was not stable over time. It means my signal, my MFI, moved farther than 2 times standard deviation for does at least one parameter. And if it does for one parameter, you remove all that section for all the stuff. Okay? Something that in the paper, they didn't look, because they only look at time versus forward scatter, side scatter. So you just see basically the flow rate and see if you get a air bubble there. But they didn't look at the MF5 variation. Okay. I trust the algorithm, because that's correct. That if you move too much in my signal acquisition, probably you get a laser delay change because of a clog or something. So that's where it goes. And you remove 5% on the dynamic range. That means events that were either in the lower or upper part of the dynamic range of the instrument. And here, didn't remove almost anything in the open range, but a lot of things in the lower. Okay. If you disagree with the dynamic range, you can just remove it. Just rerun flow AI again, just remove the dynamic range. If you think that something is wrong, maybe try both and see there's a real difference. I'd rather have lower number of cells, but good quality of cells than a big number of cells with crap. All right, so I'm gonna keep everything, and I'm just gonna run this flow AI on all my sample. And I just have to click and drag that on all my sample on just my sample list. And same thing, I need to have elevator music for the time that we wait. Or I can just cheat and open the experiment already done earlier. All right. Yep. Yes, and that was going to be my next step after that. Yeah, so we're gonna take the good event and we're gonna remove all the unwanted cells or unwanted event that we don't want. So we're just gonna do a forward scatter, side scatter, double discrimination, live dead marker. Here I'm gonna stop there because they're really looking at everything. But if your interest is in CD4 T cells, maybe you just go CD45, CD3, and really only select the CD4 and do your dimensional reduction and clustering on those CD4. All right, so done for one sample. So I'm just gonna proceed on this sample while the other one still calculating. All right, so I'm just gonna select my good event, so double click on it. Now I have only the good event there. And as Sarah asked me, I'm just gonna start drawing my gates. So we're gonna select our cells, or everything that's not debris. Okay, just call it cells. Double click. Just gonna go for the double discrimination, forward scatter A versus H. The diagonal there. To remove 100% of the diagonal of the doublet, I'm gonna do a side scatter A versus side scatter H. If you just do only one, you're gonna remove about 98, 99%. Because if the cells are perfectly on top of each other, you're gonna not see it on side scatter, you're just gonna see it on forward scatter. If they're perfectly aligned with the laser, you're not gonna see it in a forward scatter, but you're gonna see it in side scatter. That's why you have to do both to remove 100%. Side scatter, we can go to the live dead marker. I don't remember which one it was. Yeah, live dead. Okay. That's good. Select the live. Okay, my live sample. Can rename this one just as. All right, so I did my cleanup. I'm gonna stop at live here. And they look at CD45 positive and negative, so I cannot remove CD45 negative. I'm just now gonna select, done running on all of my samples. So I'm just gonna select my manual gate, included the good and bad event, and apply that to all my samples. Right. And FlowJo will know that FlowAI good event and bad event is run by the algorithm. And so it's not going to change the percentage. It's going to take the run from the algorithm. All right, so labeling, here it's only 4 samples. There's no reason to label. There's nothing, no condition, no positive, negative, or treatment or anything. But if you want to label, you just go to keyword, add keyword, and you can add anything. So I'm going to put a sample, and I can just label them 1, 2, 3, 4. I can just create a value series if I want. 1, 2, 3, 4, automatic. Okay, delete that. There you go, 1, 2, 3, 4. Okay. Label my sample. For you, just label it based on condition, maybe unsimulated, condition 1, condition 2, condition 3. Just label them there as well, if you haven't done it on the instrument first. All right, so now I want to concatenate, group all those samples together. So I want to select all my live sample. So I'm just gonna go in edit, select equivalent node, so I don't have to select manually one by one. See, all my nodes are selected for live, for all my sample. And I'm just gonna either go in file, concatenate, or I like to do a right click, concatenate population. Concatenate population, I'm gonna go in the concatenation tab, select, I want FCS file created, where to create it on my desktop. Usually you want to normalize, so make sure that normalize the number of cells between each condition. So here is the down sample. I don't like the word down sample because people always think that we remove cells. I just want to make sure that one condition is not going to overpower my algorithm. So I want to make sure that between my unstimulated and stimulated, they both have the exact same number of cells. So I'm just going to down sample here, or normalize the number of cells for each sample to the maximum number of for that sample. So that's the number here. I can decrease that number if I want, but I'm going to leave it there. Parameters is going to create new parameters. You always want to select compensated, especially if you run on different days. That's a good habit. And in the advanced option, you can rename it. So concat file, call it something else, to whatever. And here in add additional parameter, that's why I'm going to add my keyword sample that I just created, or any keyword that you created that's going to allow you to separate your sample after you concatenate them. So here's my sample. Okay. And click concatenate, and it creates a concatenate file. Here it is, my concatenate to a file right here. I can add it back to my existing workspace and close. I have my concatenate file now here, right there. Okay. Great. Into new group, concat. Green because I like green. And Kelly said this morning green was the best color, right, for Flow, so maybe that's the reason. And put my concat file in it, just like that. I don't see anything else. I don't need FlowAI. Just gonna have to wait that it's done running. All right. And now my concat file has all my sample in it. They're already cleaned up. If I look, I have 2 new parameters. First is a sample ID, just 1, 2, 3, 4 that were assigned based on the order in my workflow, but also I have my new keyword that I just created, and for you will be your condition, which was this sample keyword right here. Okay. And now you can get back the sample. So because I have my first sample, it's still one file, but I can get back and say that's my sample 1, 2, 3, 4. Yes, I remove, I delete it. Okay, so I have my sample 1, 2, 3, 4, so I can then get them back. And when I do the analysis, when I'm done the analysis, I can just show sample 1, 2, 3, and 4, or the different condition in my analysis to do the visualization. All right. We're gonna run dimensional reduction. So, can run UMAP, T-SNE, PackMap. I'm gonna use T-SNE, it's native. I'm just gonna run T-SNE, select the parameter I want to use. So it's everything except the live dead because we already removed the dead cells. So I don't need that parameter. I need back here. Yeah. And after you have different T-SNE that you run. So T-SNE, I think from 2002 to 2019, you have like 15 paper on T-SNE, on T-SNE in general. So after you just different parameter. For us in FlowJo, we're focusing on OpSNE with the optimization of T-SNE and FitSNE. If you want to run the regular T-SNE, you can just select manual and you will run the regular T-SNE. FitSNE is a fast iteration of a T-SNE. Just make it faster. We have another way to make it faster. Is instead of the KNN, instead of using exact, you can use the random annoy approximation. And I'm sure everybody in this room already used that without knowing. That's what Spotify use to select the next music that you're running. Just make it fast. All right, so to make it faster, because I don't think you're tired of the music between each time, I'm just gonna run the annoy and FitSNE, which is gonna make it as fast as possible. When you run T-SNE, OpSNE, sorry, you don't have to change the iteration number. You don't have to change the learning rate. Those two numbers are determined by the OpSNE option. The only number that you need to change is the perplexity. And the perplexity, to make it easier, it goes from 5 to 50 usually as a number in Flow. In Flow, we have more in 30 to 50 range. And it's the number of cells. One cell is going to talk to how many cells. So usually 30, 40, 50, depending on the number of cells. That's what you want. I'm just going to leave it on 30. You see, with running algorithm, you have a lot of coffee break, which can be good. Or a lot of things to do other things at the same time. Not spending your time. No, Siri, I don't want to do anything. Thank you. Usually that's because of Zoom. I realized that doing live, like virtual training, when I'm on Zoom, is taking way longer. Zoom is taking so much memory on your computer, and it's crazy. And Google Chrome also. And I was really surprised that those two programs are using a lot of resources from your computer. Yeah. Yeah, we're looking at 40 parameters, a lot of cells. I really didn't downsample, so you have 1.3 million. Oh, it's coming there. Yes, you can downsample as much as you want. After, it's really depend of your frequency of your cell population that you don't want to downsample too much because you don't want to end up with one cell. So really depend on your question and your cells. And also really depend on your computer you're running. It's laptop, it's not, the best computer ever. 16 gigabytes of RAM. i5, I think that's the minimum required for FlowJo. So I don't know why they don't give me a better computer, but another question. it's taking too long. I can cancel. I have it run already. Getting there. No, no question. Sorry. In my hand, it's the other way around. Usually. Yeah. I don't really know why is, but yeah, in my hand, it's usually slower to run UMAP than TSNE. No, you can run that. No, it's just a different way you run TSNE. So you just, when you do this type of analysis, as long as you mention what you've done, is correct. And funny enough, in my hand, I know it's better than the Barnhurst CNN number. It's faster and it seems like better in my hand. I'm not gonna claim it, not gonna publish about it, but I usually like the Hanoi better. I put all the file in one, and after I just put different keyword in it. Yeah, it was. Yeah, the questions was either I do different workspaces for different analysis, or just concatenate all the file into one and just use the keyword to be able to separate my sample. Here's the keyword. Yes, you can have hundreds of thousands of cells sometimes if you have a huge experiment or plate and you run, but at the end for the algorithm anyway, you're gonna have to concatenate everything in one. That's why downsampling may come in play if you're not looking at a really rare population. Correct. Yeah. So you have to do two ways. So if you save as a workspace, yes, you're gonna have your workspace here, my FlowTex workspace here at the bottom, and my FlowTex folder. Those have to be together to be able to analyze the data, because everything about the algorithm or the R script will be in that folder. If you want to save it and move it or give it to someone else, just save as an archive instead of saving as a workspace. When you save as an archive, everything's going to be embedded in one file and you can share that. As long as you have the folder at the same place, usually we can find it. The software is smart enough to find it and it should be able to reconnect, but yeah, it can happen. We're in the iteration, so getting there. The worst thing is we're waiting for something that I'm gonna cancel in 2 minutes, but I think so. Just getting there, but it's gonna take probably 5 more minutes. I'd rather, keep moving forward and spend time on really showing the data than just waiting for the algorithm to calculate. not the right FlowJo. All right, so just make sure to show you that's exactly the same. I run my flow AI here, select it, concatenate my file. That's the concat one right here. I've created a group to concat one and have my tsni run. Okay, so that's my UMAP. And I have the tsni that was run as well. Doesn't matter which one you're running. Here's my tsni. Okay, so if you compare to the paper, that's UMAP. If you want to see exactly the same thing, you can go UMAP or really similar at least. My UMAP, so really similar to what they got there. So when you have your visualization, now you want to run your clustering algorithm. So in the paper, they run FlowSum, so I just run FlowSum on it. Same thing, select your parameter, go in Tools, Workspace, Plugin, FlowSum. Select your parameter, everything except live dead in this case. You're gonna select how many clusters you expect to have. So that's kind of the downside for me for FlowSum. Usually you don't know how many clusters you expect. So it's better to create too many clusters and merge those clusters than creating not enough. So my rule of thumb is the number of parameters I have, this number of clusters I create. So the 40 color experiment, I'm gonna put 40 cluster in FlowSum. For that, they do a sum grid to be able to find those clusters. If I create 40 cluster and the grid is 10 by 10, that means 100, it's not gonna be much there. So I need to increase that number to something that makes sense a number. So usually I put for 40, I'm probably gonna put 20 by 20 or something like that to create. And after just the visualization, what do you want to see in the pie chart? In the spanning tree, do you want to see pie chart or a specific parameter as a visualization? You also have some advance as well to choose the color. If you're colorblind or you don't like the default color, that's available. So I run FlowSum, same thing. We don't want you guys to wait too long. And we had our 37 parameter here in this case. 36. And of course, we could have done it manually. Okay, so we did the gating manually the way they did it in the paper. I reproduce exactly the same gating strategy with all the color and everything. So you can really have it paper ready within FlowJo. You don't have to go to Paint or whatever software to get all those boxes. Everything can be done in FlowJo. All the text can be done, the arrow and everything. But what matters to us is really the visualization. So here's we have the UMAP. I can right click, go make multi-graph overlay, go N by N plot, select the parameter that matters. So not N by N plot, the multicolor color mapping, there you go. And select the parameter that matters. Maybe those are too much. I don't want just something wait too long. And it shows me the expression of those markers with a color coded blue for negative, red, orange for positive. I can see where are those markers located in which cluster for tsni or UMAP, depending on what you use. So that's good for, let's say, presentation, how to present your data, how to talk about your data. Maybe not good to really define each cell population. So to define each cell population, we did the FlowSum, and I did overlay just my FlowSum gate into the tsni or the UMAP, and just to make sure that everything was covered up and no population was missing. And to my surprise, there was one population that was missing. There was something when I overlay my manual gate on my tsni and UMAP, there was a cluster, an island, that was not defined in my manual gate. And I was like, how come? It's been 40 color on my immunophenotyping on blood, and there's something that's not defined. And it's not defined, if I remember correctly. Look, where is it? Is the black color thing here, the ungate was really well defined population. I think that's right here, something right there in the center. Okay. I was like, all right, so it can happen. You know, that's usually why we say manual gating is elusive. And maybe there's a population that's never been defined in the field, and I may want to know exactly what that cell population is. So an easy way to do it, I just open my Disney. Okay. And I did create a gate around. So a query gate, oh sorry, it was on top. A query gate around this cell population saying there's a well-defined island. That means a well-defined cell type compared to anything else, and nothing is defining that population. So I create a gate around it, and I'm gonna try to define that gate. So I just put my T-SNE there, overlay my query gate on it. So that's my blue thing here. And I just right-click on my T-SNE, make multigraph overlay, histogram. I can look at all the histogram for every parameter. It's small. I'm gonna make it a little bit bigger for you. And you can see this one is really present in my sample 1. Absent sample 2, a little bit present in sample 3 and 4. It's negative for most of the marker, except one marker, if I remember correctly, down here is CD39 with mid expression for CD39. And everything else, if I go down, everything else is negative. That's, that excited me yesterday. I was like not sleeping, trying to prepare that. And I was like, wow, we found a new cell population, CD39 positive. So CD39 positive only. So I didn't know what CD39 was staying for. So I went online, look on PubMed, got excited. And what is for CD39? So it's a ADP, ATP, AMP adenovirus pathway, CD39 with a novel checkpoint. Inhibitor is usually for CD8 T cells, but they're not CD8 positive, so that doesn't work. So go down. What else? The Treg. And basically, I was looking and didn't find anything that tell me what's CD39 positive on. So now I want to solve them, right? So that's why hyperparameter analysis and bias analysis can help you, even if you think that all your manual gating is correct. Okay. Of course, I'm not gonna pursue that. I don't have the data, I don't have the sample, but it was exciting just by something that's been published and that you think you define everything manually, we can still find something that can be maybe interesting to explore. All right, so that was the comparison between manual and dimensional reduction. We use clustering algorithm just to define now each cluster, and I want to define each of the cluster. I don't want to call it flow sum 0, 1, 2, 3, 4. I want to define those clusters, and to define those clusters, I'm just gonna use Cluster Explorer. And Cluster Explorer, I'm gonna select the parameter I want to visualize, everything. I can remove the live dead. And I'm gonna want to see my UMAP, T-SNE, and of course, my clustering algorithm, flow sum. Click OK. Gonna have a little bit of time to get all the graph together. It will not be too long. All right, so zoom out. All right, so I have my T-SNE, my UMAP, Okay, here visualization. I have my clustering size. So here I see in percentage. I can remove the normalization and just see the real number of cells if I really want for each cluster. I have my control panel of Cluster Explorer. I have the representation of each line is one cell type. I have the heat map as well. So I can start gating. So here you see on the line here, I see basically the baseline of the graph of 0, 10 to the 2, 3, 4, 5, 10 to the 6. So I can really gate based on the graph. If I have the doubt on where to stop, I can always open a 2D plot and look for it on this to know with my manual gating where will I stop. I will have stopped here, maybe at 10 to the 3. I can draw a gate at 10 to the 3 on my line right there. So let's look a little bit at the data. I'm gonna look here, see the data in mean. I can see it in median if I rather. Okay. You can have the scale value, which is the real true value. If you want to bin the data, you can select the channel value if you want. The thing with the channel value, you don't get below 0, but I like to get the real value, the scale value. And I can say, now I can select by drawing gates between my CD8, for example. I can gate all my CD8, okay? And it's gonna show me all the cluster that are CD8 positive in the heat map, also with the line in my dot plot, my t-SNE UMAP, and in my cluster size. I can really define, okay, so all the CD8 positive cluster are those cluster. I can create an AND gate, create a gate on CD3. I can find the CD3 now somewhere. Halfway in the middle, CD4, CD3, I see here, okay. All CD3, create the AND gate on CD3. If I want to create a NOT gate, I can select NOT gate and select NOT gate for that. And I can really define all my cluster and exactly tell if that, whether positive, negative for, and say my cluster 1 is this type of cell, my clusters 2 is this type of cell, etc., etc. If I have many clustering algorithm, I can change my clustering algorithm from one to the other to compare, or I can also select based on my keyword. So here's only my manual gating, but if I had creating condition, instead of saying for all the concatenated file, I can select which condition and have the visualization only for that condition. All those graphs, all those information can be copy and paste, and saved for presentation. If there's any value, so for the heat map, and there you can even copy the data and export it directly to Excel without any issue. To compare the data between condition, let's say I don't have condition here. Let's see if I have keyword only on there. If I do that, I'm gonna have to rerun. Up. Wait, pick on the histogram. Okay, I can create pick on gate for my condition or my sample, and after I can visualize based on the sample here in Cluster Explorer and see which cluster affect which result. All right, so let's say we define a cluster that's cluster of interest. That's really your part here in defining all those population. And after we define the population, we just go in plugin, Hyperfunder. You can select which number of gates dimension you want to create. You want to select if you want to improve the purity or improve the yield. So one, you put exact same value for the yield and the purity. If you increase that number, you're gonna increase the yield. If you decrease that number, you're gonna increase the purity. And after, you can choose the type of gate to create, either polygonal or rectangle. In this one is a beta version, so you have this new polytop version. The author of this plugin just updated us. It didn't tell us yet what's polytop, so I cannot help you there. And you can select the parameter you want the algorithm to take in consideration to create this population. Of course, you want everything. And after you select the population you want to sort, so you can, of course, select one, or you can select many, depending on, if you have S6, you can sort 6 populations, so you can define those 6 populations, click OK, and it's going to run the hyperfunder and find the population there. I didn't click OK. Did I? There you go. So it's going to try to find the cluster I define in the shortest number of gates as possible. And at the end, it's going to show it in FlowJo. You just have to right-click on it, export to Diva, and you're ready to go on the instrument to sort that population. I think we don't have to wait, and I take any question. Thank you. Two cells? No, there's no minimum number. After, if you try to define your population, you need to have a specific, at least enough cells to define your cell population. Same thing for, usually it's the highest number of cells that can be a question. If you have too many cells, if you go to 100 million cells, you're gonna need a really powerful computer to run, or it's just gonna take time. So sometimes just run it before go to bed or before your lunchtime, and it's gonna run overnight.
Nick, question about the use of Flow AI. In our hands, what we see is it can be too aggressive, and we kind of saw it. I mean, if the quality of the sample it's low, and that is what it is, and you need to get rid of bad events. But when comparing with other algorithms that do that cleaning, the amount of events that you get removed, comparing the same data set, same sample, it's lower. And I feel that in this data set where you have over 600,000 events, sacrificing 30%, it still gives you a good number of events for the analysis. But with samples with low cellularity, 10,000 events, then getting rid of 30% can be important. So wondering if there is any incorporation of other algorithms that are less drastic because I know you can adjust the tests that are done based on intensity or range, but just wondering if the plan is to include others that are less impactful.
So one of the oldest algorithm is FlowClean. We have it, and yes, it's way less aggressive than Flow AI, but FlowClean only look at the differences in the flow rate, not really in the dynamic range. So after, as I said, I'm not gonna say run Flow AI on every sample. I know my colleague even like FlowClean better than Flow AI. It's just my personal preference for Flow AI, but I agree, it's really drastic. It's gonna remove a lot of events most of the time. Peacock's supposed to be better, slightly better. And even in the Peacock paper, they compare FlowClean, Flow AI, and Peacock. And FlowClean was way lower in the ranking in the algorithm for removing unwanted event. Flow AI without the flow rate and with the flow rate was really similar. So I was really surprised that the value were almost no difference that if you were using or not the flow rate. And Peacock, of course, because it was their paper, that was the best. But it's not perfect tool. I think, we know that for normalization, cytonorm works, but not on every sample. So I think there's a way in the field that need improvement in all those algorithms, especially for the cleaning the sample in general, normalization, cleaning.
Yeah, so I guess it's important to leave that message out there. That
Yeah, and after you
comparing
run it, try it, see what you removed. Do you agree or not? It's always you're the scientist, and so you make your own decision. As long as you, as I said, there's no good or bad like way to do it. As long as you document what you do, I think everybody is clear and can reproduce it.
I had a question about overclustering. You had mentioned, that's one thing you have to put into FlowSum is defining the number of clusters. And you said it's better to estimate the number of clusters than underestimate. Is there a simple solution in FlowJo of merging clusters after the fact?
Yes.
Oh, how do you do that?
You can merge the cluster within Cluster Explorer. You can define the two clusters and say merge. This cluster is going to create a new cluster merge. In a Cluster Explorer, you can select the two clusters and say merge cluster.
Okay, thank you.
Right now you're doing manually.
I was just going to ask for a suggestion. So, I know the FlowSum doesn't do, like you do not know how many clusters to start with when you're running the FlowSum, but do you think running an X shift, because it is actually going to come up with a cluster number and then go to FlowSum and say, hey, plus X shift is giving me 18 clusters. I'm going to start with 20 clusters. Will that be a good approach rather than like doing a whole lot of clusters?
There's a lot of users does it, and I think that's a pretty decent approach to go to Phenograph or XShift, someone that's going to give you the number of cluster, and after just run FlowSum for the advantage of FlowSum that you can add sample after on FlowSum. That's the only algorithm that you can do it. And having the visualization also for, it's nice to have the spanning tree on your paper. So instead of something else. So yeah, that's one way to do it. You just spend twice the time of running it. So it just depends. Thank you.
We're going to have like a 15-minute break. So we'll be back at 2:15 and a couple quick announcements. If you need parking validation for the garage, the UT Center Tower, you can go to the registration table. And then it looks like somebody lost an earring. So if that was you, you can also go to the registration table. Okay, thanks, guys. It is shared, but I'm just trying to minimize these guys. So that yeah. Them too. Okay, sorry about that. We're back. So our next speaker is Robin Kamucheka. Is that right? Nailed it. Okay. Robin graduated summa cum laude with honors from the Honors College with a BS in biology from UT San Antonio. She brings many years of flow and image cytometry experience to de novo software. She has designed, executed, and analyzed multiple experimental protocols for blood samples generated from human and animal research. Prior to cytometry work, she studied chromosome 18 abnormalities and genetic expression in humans and plants. Now at de novo software, Robin uses her unique background to partner with FCS Express customers to meet their data analysis objectives. And we're very excited to have her talk about basically showing a very similar kind of thing that we saw from Nico, which is the FCS Express workflow, working on high dimensional analysis. So thank you, Robin, for coming, and take it away.
First, I'd like to say what a pleasure it is to be here with you guys today and just be able to present some information about, there we go. Give you some information about FCS Express and, perhaps just discover some things you didn't know about FCS Express and maybe try it out if you get a chance. So today I'm going to kind of go a little slower. We're going to talk about how to demystify the complexity of creating a high dimensional analysis strategy. Sometimes you look at your data set and you're like, oh my gosh, like, what am I going to do with this, right? We know that Flow has now built the capability to gather all these parameters and collect all of this information, but sometimes it can be really hard to start dissolving that information and getting the importance of what those things mean as a whole over the whole sample. So let's go ahead and just jump right in. As we're collecting those different parameters, it can be a blessing and a curse, right? If we don't know what to do with all of that information, we can invest a lot of time optimizing the experiment, then we get to having these data files and we don't really know what the next move is. So what I'm going to do is I'm going to address some of these challenges that we get into, things that I see with customers when I'm working with them with FCS Express, and kind of meet you in those places, and then go through the software. We're going to go in real time and look at FCS Express and see where we can meet those challenges and overcome those challenges using the software. So starting with our first challenge is bivariate plots provide limited information. If we're looking at 40 different parameters, it's going to take a whole lot of plots to look across all 40 of those parameters. And while we're looking across that, it can also be hard to deduce what's happening in between, right? So I'm looking at these two parameters, but what does this mean to this plot? And where do I go with that? So we're going to start looking in software here. That over. And first, I'd like to acknowledge where this data is coming from. So we're coming from, thank you. So we're going to acknowledge where this data is coming from. So we're using the optimized multicolor immunophenotyping panel 69, which was developed by SciTech. And this is the information that we're using to kind of look at high dimensional analysis. We do have the capability in FCS Express to bring in data from any commercially available cytometer, as well as image data. So you're not limited to just one data set here, but we're just going to explore this. So let's look at our bivariate plots. So on this page, we're going to see all of these different plots that are demonstrating two parameters, right? So 2 by 2, we're looking through all of these plots, and we can see we've got kind of a lot of them to just collect information about each of our parameters. Now, one of the cool tools in FCS Express is that you can select a plot and there is a multi-plot tool. So you can build these really quickly, really easily, so you can look at these relationships, but still gleaning the information of how the relationships work and what this means as a whole can be really difficult. To confound this, we also have the problem of manual gating. We introduce our own biases from our knowledge, from our experience. We also just, sometimes we have gradient expression. Where do you put that line? We have some tools like FMOs isotypes if you still use that route. And we start setting those gates and do our best to remain objective. But there's always that opportunity for error or the accidental, we've made a gate too shallow, and so we're missing information. So what we can do is we can use our bivariate plots in addition to the high dimensional analysis strategies. So it's not an either or. We don't say, oh gosh, bivariate plots are not working for us. We still utilize them, and we utilize them as a tool to confirm that when we're looking at our high dimensional analysis, maybe a UMAP or TISNE, that we can see that our gating strategy, the formations of regions of populations are kind of aligning. So you can kind of have a two-way conversation with your data to make sure that you're getting very accurate results, and you're not just kind of, oh, wow, there's a whole new population. Where did that guy come from, right? We want to make sure that we're solid in what we're analyzing. Sorry, a little off here. So then we go to our next step, and this is really where the rubber meets the road. This is where we start working with our data management, and we want to make sure that we're pre-processing our data, that we're handling our data correctly. Sometimes it's really easy to get those data files and be like, yep, data files to TISNE in 2 steps. But that's not always the best strategy. Sometimes we need to slow it down a little bit and really take in what's happening with our data. So there are some things that we can do in FCS Express to make that very accessible to you. Merging data files, cleaning your data files, determining scaling, normalization, downsampling, and parameter removal. So all of these individually can be very important to your data, right? We want to make sure that we are putting good data in so we get good results out. That's the line is always said in flow cytometry, garbage in, garbage out, and it remains true even for high dimensional analysis. Sometimes we think we can kind of blur that line because we're doing populations and there's a little bit of space to, kind of flex a little bit. But it's really important that when we come back to the basics, that we are definitely getting good data out. We want to make sure that we can rely on the information that we're conveying to others about what we found in our experiment. And we want to be able to answer those scientific questions with as much certainty as we can do in research, right? Because we always have that room for some error, maybe not understanding what we're seeing clearly, but we want to make sure that we're not just making things up as we go. So some of the tools that can be very helpful is we can add metadata. So these are adding additional keywords to your data set to help group or combine your data sets to give you additional information about what's happening. This does not change the actual FCS file itself. You create a new file that's imported as if it was part of the data file, and then you can use that in any analyses moving forward. We do have the ability to merge data. This is great because when we're doing those high dimensional analysis tools, we want to make sure that we're creating one structure and then looking at different populations and how they're contributing to that structure without jeopardizing the overall structure of that TISNE or UMAP. We want to have that one visualization and then look at how things are contributing back and forth. Now, data cleaning is a big deal. Sometimes we skip this step. We're like, oh, it can't be that bad. There's not too many outliers. It's, I did really good. It was a really clean experiment. And yeah, we can collect the best we can, but light scatters, light does what it does, and we can't control all of those factors internally in the instrument. So data cleaning, what it does, it's going to remove those outliers, the debris, the aggregates, the things that can really start skewing your data if we have too much of it. We want to, again, make sure that when we're bringing this to our high dimensional analysis tools like TISNE and UMAP, we're not getting something skewed or finding new populations just because you have so many aggregates. We also have the ability to do parameter scaling. This really becomes essential to your data set because we want to make sure that the lower intensity expressions and the higher intensity expressions are getting the same weight when we start using these algorithms, right? Because like if we move from 10 to 20, that's a twofold difference. Now, if we do 10,010 to 10,020, that can feel like a different weight if we're going with like twofold difference, 0.001, right? So we want to make sure that when the algorithm runs, it's recognizing that these are still the same values between the things. So scaling can be very crucial before you run those algorithms. Normalization is a bit finicky. Some people really love it. They say that it can help benefit between variability between the parameters, variability between samples. But if you're not careful in how you're using it, you can actually also remove the variability from biology. So it's a very careful balance. You know your experiment. You are the expert on what you're doing. So just be mindful when you're looking at these samples to notice like, like I know when this patient is definitely expressing something different from this patient, but when I normalize this data, suddenly I'm losing that difference. That could be a very valid biological difference, and you don't want to remove that from your sampling, because what we do with the high dimensional analysis is we really exploit those variances. We want to see what is the same, what is the difference, so we can really get a whole picture of what's happening. Downsampling can be really great. There's a bunch of varieties of downsampling that we have available. Gives you the ability to run your algorithms quickly, can still give you accurate results about what's happening with the data file itself, but then, gives you the ability to also start sliding. So if you know that you're looking at a rare population, you can actually use weighted densities or other types of downsampling, targeted densities, so that you can skew to actually collect those rare events. So now you're actually seeing those rare events. You can bring them out of your data and really bring them to the forefront, where if we just do maybe interval downsampling, we can actually lose those events. So it's really important to know what your scientific goals are, what direction that you're going, and how to use the tools that are in front of you. Parameter selection is also very much related to the same thing. We want to make sure that we know what parameters to select before we start doing our high dimensional analysis. Sometimes it's great, we can select them all and we can create a t-SNE and we can start looking at different populations and how they're showing up. But at other times, we want to really dial that down, look at a specific population, and then see how that changes when we're looking for certain features or expressions of markers. Now, the last one is about parameter removal. This is just a way to manage the parameters when we're working with our high dimensional analysis. When we scale the parameters, we'll get new parameters that say scaled at the end. It can be great while you're working in the pipeline, but then when you go to your plot and you have 16 extra parameters to manage, it could be a little bit more painful. So that's a feature that is, again, available, not always necessary, but can be useful. So now we're going to move back to the software here. I can get back. Oh, okay. It will not let me out. Right, we'll go that way. So let's talk a little bit about starting to implement some of those tools that we were talking about for data cleaning. So when we go to the Tools tab, we're going to find our transformations. In this transformation window, you'll see that it's docked to the side. Most of our pop-up windows are going to be dockable, just so you can create the space so that you're not having to worry about managing the space on top of trying to figure out what to do with your analysis. So we can dock it on the side, and then we can start building this. Pipelines are really a powerful tool. What they allow us to do is stack and go step by step on how we're going to handle this data. How are we going to approach this? So it's kind of like building blocks. If I'm going somewhere, I know that I need to get in the car, buckle my seatbelt, start the car, go here, drive to wherever, right? Those are the steps. This is the way that you can shape this. Now, these are all saved with your layout. So once you create this, you can save it as a template, drop your new data files in there. You'll make some adjustments to your gates. You'll come back, but then you'll get to spend that time optimizing this, these transformations to make sure, again, that you're getting those accurate results. So expanding this pipeline a little bit, this first step here in our pipeline is kind of the biggest part, right? Because this is our starting point. Whatever we start with here is going to be available downstream. If we exclude something here, it's not going to be available downstream for us. So here in the pipeline, you'll see this new classification folder. Now, this is a helpful tool if I'm starting from a merged data file, because then I don't have to unmerge it. I can start with that merged data set, and I can tell the software to parse out each data file individually by using this classification identifier. So it tells the software, hey, I need you to use this classifier identification and run these things on each sample individually. This becomes really important for like new interval downsampling. I want each sample to be handled exactly the same. I don't want it to be variable when I bring in each data file. And then for the Flow AI, it becomes even more important because we're looking at keywords. We can use keywords within the data file to start using these checkdowns and how we're reducing the data, how we're excluding those outlier events. So here we also will always have multiple variables that you can control how strict or how loose you want to start cleaning up that data set. So this is our flow rate check. What it's doing is making sure that we're kind of have a standardized flow. We don't want to see big gaps. We don't want to see anything getting really wonky. We're going to exclude all of that so we know that we're looking at good data. We're getting a good flow. We're getting good signals. The signal acquisition is exactly the same. We want to make sure that we're not seeing huge jumps and spikes with each parameter. We want to make sure that we're kind of getting this clean, oh, okay, this is the sweet spot. This is where our signal's showing up. And same with our dynamic range, right? This is where we start using those keywords, and these are keywords from each data file. Now, if you didn't have this classifications folder, you'd need to run these steps individually on each file. There are some preferences. Some people like to run these first on each data file, export that data file out from FCS Express with that transformation, with that clean, and then merge them after. It's very preferential. We have a lot of ways to merge data. So one of the ways that we merge data is just virtually. We can bring it into the workspace by clicking on this blue plus icon. And then here at the bottom, you'll see that I've already changed the file type to merge these FCS Express or these FCS files. We have plenty of different files types. So if ever you are in the software and you're like, FCS Express has eaten my data files, which I've heard happen, it really comes down to this tab here. If you just check this and find the right file type, you'll see your data files and be able to bring them in. But once we merge the data file, it's going to show up in our data list, as you can see there in our 5th data sample. This is actually a merged file, and that's why let's see if I can grab that there. That's why I use that classification folder. It's just easier to work from a merged file for this instance. But again, if you did need to do this a different way, you wanted to treat each file individually and then merge them after, we do have on the data tab the option to batch export your data. You can select this bottom one, this bottom radio button here, and then export as a merged file. So there are multiple ways to skin the cat in FCS Express. We kind of want to give you the flexibility to either create a new data file or just work from where you are in the workspace. All right, so now we're going to pop back to that PowerPoint here. Right. And now we're going to start talking about how we're going to start building up and developing a high dimensional analysis strategy. I know you're like, but Robin, we just have talked about all of this pre-processing and what am I doing? Once we start getting to this point, we've done all the work to clean up our data to make sure that we're going to get something good out. We have to really focus on that scientific question. What is it that we need to know or want to know about our data set? There are so many different algorithms that we can use. There's a lot of information about all the algorithms. We all look at publications every day and we're like, oh yeah, look, there's a TISNE, there's a UMAP, there's this, there's that. I want to do these things, but we have to understand the why. Why do we want to see? What are we trying to get? What is the information that we need? Additionally, you'll have the option to select a population of interest. So whenever we start building these dimensional analysis strategies, we need to define like, where do I want to start? We can start really broad and be like, I want to see everything. I just want to look from the live cells down and see how things start populating and spreading out. Like, what are the same? What are the differences? What are the relationships? What does that look like? We can also get very targeted. We can be like, hey, I want to look at T cells, and now I want to look at specific features for these T cells and see how they parse out and how these populations are built within a certain subset. So having those questions, knowing those questions up front is going to be one of the most powerful parts of your analysis also. Being clear so that you have that objective. Again, just like getting in that car, I need to know exactly where I'm going and I'm going to drive in circles. And trust me, that has happened to me in analysis plenty of times where you're like, I don't even really know what I want to do. So I just want to explore. That's valid too. Sometimes we don't really know what our data is trying to tell us, and we're kind of trying to explore and research it. That is also a valid way to use these algorithms. We don't also, sometimes we just are exploring new things. right? We don't have any information. There's no publications. We found this population and maybe it's something or maybe it's not. We kind of have to take that into consideration. So here are the algorithms, more of the most popular algorithms, I would say, that are used in FCS Express. This is just a list. It's not complete. There's more algorithms that we can use. And I did just want to take a moment that if you are a newer user to FCS Express, I don't want you to be like, oh gosh, that's not for me. We have some websites already built for you to look at high dimensional data analysis. These are videos that are going to walk you through the parts. So the pre-processing that I was just talking about, scaling, normalization, downsampling, using different algorithms. We have plenty of videos that are going to help walk you through this experience. Also, I will reiterate that your best resource is going to become to one of our task team members. We do screen shares and make sure that, you know, while you're working through your analysis, you have the support. You're not going to have to just be like, oh gosh, here we go. One more thing to figure out. Flow cytometry is hard enough, so we're going to be there to support you. Now, moving back to the software, we're going to go back into that Tools tab there and look at those transformations. Now, this is pre-built just for time sensitivity so that you have some visuals. We're going to make something that's short and sweet also so you can kind of see how the software is going to run and how it's going to handle things. But I also just wanted to kind of give you a bird's eye view of some of the things that you can accomplish. Oh, and there we go. So after we've done the pre-processing, remember, this is like building blocks where you can use information that we've used in the previous steps and carry it down to the next steps. So here, after I've done all of my downsampling, I've done some data clean, I'm going to start working through my FlowSom. So as you can see, it's right here at the top of the list. This is a predefined algorithm. So when I create these pipelines, I get this second little drop-down menu. And here you're going to find a ton of different algorithms that you can use and implement in your experiments. So predefined algorithms, you have a nice list. We have FlowAI, FlowCut, FlowSom, Phenograph, Spade. Downsampling, here's the multiple different types of downsampling we were talking about earlier. Dimensionality reduction, those are favorites. Clusterings, also favorites. These are actually breakdown pieces of the FlowSom that we saw earlier. So if you just wanted to use parts of that, you have that flexibility. You can just do pieces. We do have visualization. So if there's a specific way you want your data to be portrayed in a plot, you can adjust that and make sure that you're seeing things exactly how you want things to be represented. We do have this mathematical tab. The 0 to 1 scaling is a way we can normalize. We have normalization, scaling, simple parameter math. Then we have the quality control, again, parts and pieces of the FlowAI that we saw earlier. And then some random parameters, feature extraction, and then miscellaneous. I will take a moment here for my fans that are R script and Python ready. That's not me. One of my favorite features of FCS Express is the fact that I don't have to have that information. We've built everything in the background, so you'll need just a few items of like, how many neighbors do I have? How many iterations? How many times I want to run this algorithm? How closely clustered do I want it? Those are the perfect questions for me. Anything beyond that, I'm like, well, I don't know. So, but if you are that Python person or the R person, we do have the ability to build in Python transformations. These are pre-scripted, predefined scripts that you can just load in and get some extra transformations. And we'll take a look at that slide in just a minute so you can see some great work that my colleague Andrea Valle did with this Python transformation, because he's the guy. So if you guys contact me about R or Python, I'll be like, we're going to talk to Andrea. I'll be back in just a few. If you are an R person, though, underneath that initial block where we found that pipeline, we do have R scripts that can just be built right in. So if you have some R scripts, you want to do R add parameters or cluster transformation, you can bring that right into this space and get those analysis pieces in here as well. So going back to our FlowSom, you'll see that when it's pre-built, it kind of nestles in a bunch of different steps. So here we have the scaling. Again, scaling, we want to make sure that we're kind of giving the same weight to each of our parameters. You can define what you select. If you want, you can start filtering things. So if you're working with a ton of parameters, you can be like, ooh, yeah, I'm not trying to select them all or not select any of them. You can start filtering them out. You can right click if you know, hey, I want to select the majority of these. use that select all feature, and then just start parsing out the ones that you don't want to select. You have that kind of flexibility. These are the parts that I was telling you about, just having a couple options to start outlining how we're going to define these transformations. Again, you don't have to know all of the things. We can start this in stages. So even if you're brand new to Flow, don't be intimidated. We've got some default settings and they work just as well as working to optimize them. You start somewhere and then you start building. You don't have to be a pro to get into this. You just start. So here we're going to define some of the things. How do we want to start defining these clusters? What are we looking for? And setting these options. But again, if you're not sure, start at the default and then move slowly. Adjust one thing, see how it changes your data, and then adjust another thing. Beauty of this is you can copy and paste these pipelines. So you can have multiple pipelines in the same workspace if you're doing something comparatively. So again, you don't have to have it all figured out. You can kind of walk through it. You just know, I want to go that way. It's important to me because these are the things that I want to discover. And then the software is going to help you in the background. So these other few options here are just defining how I want my clusters, how I want them to be visually represented in the graph layout. And then new consensus clustering is that second clustering on that flow SOM. So I've got my initial clusters. Now, what's the bigger picture? How are those clustered or reclustered in different groups? We also have this parameter removal. So I was telling you about this earlier. When we get those scaled or we're adding a bunch of additional parameters, it may be a little bit more to manage when we're in the regular workspace. You can remove them. But if you're going to do something downstream, like we're with our UMAP, our second part, we're going to use those scaled parameters. So I don't want to remove anything yet. So you can uncheck things. You don't necessarily have to use them. If you don't want to use them now, you can add them back later. You have a lot of flexibility in the decisions you're making. So here with our UMAP, again, we're going to grab all the scaled parameters. So they're usually checked here kind of automatically. The software figures out like if she's scaling, I'm pretty sure she's going to use them. So it will check these for you as you move and add this feature. And then at the bottom, this is kind of where if you just know a little bit, you can kind of do a lot. So I just know, hey, yep, I'm going to let the software start at this default position of number of neighbors. This is how many cells that I think are going to be neighboring my primary cell that I'm looking at. Minimum dim distance, how tightly clustered or compact do I want my UMAP to appear? And number of iterations, how many times do I want this algorithm to run? This last one, the generate new random seed, you can start at 6, that's the default. You can push the button and it will just start at a certain number in your data set. That with the importance of this is with UMAP, it's very stochastic. So if I keep the settings the same, I'm going to get the same results. So if you are going to start testing and manipulating these values, you want to keep that generated random seed the same too, so that you know you're starting at the same point, you're doing the same things, you've just modified a little bit so you can compare across the board. So now let's look at some pictures because that's really the fun part, right? Like, what does this mean? So FlowSOM is here. Our FlowSOM clusters in 2 ways. First, it's telling me where things are initially kind of parsing out. Where are the groups here? And then when I start creating those groups, what is the bigger group? Are these parts of a bigger group? And that's how we get the secondary clustering, the colors on the outside of those circles. As you can see here, we're going to have this scaling that's going to tell us whether it's expressing or not. So the more yellow-green it is, the more highly expressed it is. The more blue-purple it is, the least expressing it is. On this FlowSOM 2, I should tell you that you can create gates. So even if you're looking at this data, you can right-click on this plot, create some well gates, select these populations or clusters individually. You can actually even collect all the secondary clusters. You can automatically create those into gates. So if that's information that you need to use, with a few right clicks, you can get there and make sure that you're getting the information out. So let's look at UMAP. This is a very popular one. It's in papers all the time. One of the beautiful things about the interactiveness of the software is I can create gates. So I did this from a merged file. And now I want to start picking this apart. So I'm using that classification identifier, which is a file identifier. And I've actually just kind of assigned 2 as a control and 2 as a test. Now, if I start taking one of these gates and modifying it, so I'm going to do the test. So it's going to be that upper right plot. you're going to see I can start looking at individual file contribution as well. So we're going to see some changes as we start moving back from file and file. We can also expand it. So now I know what my test sample looks like, right? So now, okay, that's what test looks like. At the bottom, my control samples are being displayed, and I can see the differences. You'll see here in this corner here, we have a pretty clear difference. We've got a pretty strong population here, and up here, not so much. We're kind of missing, doesn't seem to be expressing the same. And then the reverse is true here. In the test group, we have this population here, and it's not so defined here. So those are some changes. Those can be important changes, right? And that gives us a place to start understanding, okay, so if I gate on that, what does that look like? What information am I going to have? And I'm going to move this gate, maybe if my mouse will let me here. And again, looking at individuals. So you don't have to just keep one gate. You have the tools here so you can start really exploring. And that's really what I find best. That's going to be your best teacher. If you're new to figuring out how to do high dimensional analysis, get in the software, play a little bit and figure it out. You're not going to break it. Worst thing that happens, you're like, oh, that went horribly wrong. I have a sea monster instead of a UMAP. And that's fine. You got to start with the sea monsters and eventually you start getting some clusters. So here we can also look at different markers, right? So we want to look at different expressions. I want to see how to change this markers. Now this is a little bit unique. We have our UMAP 1 and 2 on the parameters there. And in our legend is how we're going to change this. So similar to how our parameters change, if you click on this here, you'll see that it will bring up all my parameters. And now you'll see, I didn't remove those other parameters, how long your parameter list can get if we're not kind of managing how many parameters that we have. Also, a great tool, like I mentioned in the beginning, even though our manual gating can be a little bit flawed, right? We're not perfect. We can use those gates to start looking at, are my gates aligning with how the UMAP is, or whatever TISNI, are they aligning with what I'm seeing in this map, right? It can give us some ideas like, ooh, that might have been the best gate, Robin, maybe I need to clean that one up, or vice versa. It could be like, wow, this is really getting the information and separating the information really nicely. So this is the best optimized presentation I can give. I'm going to move back to our PowerPoint here, which clearly likes starting at the beginning. Sorry, guys. All right, so these are those Python scripts that we talked about that you can bring in to FCS Express. It is a plugin. These scripts are already written, so you don't have to know that much, which again, makes me very happy. But these are some of the additional high dimensional analysis tools that are available through that Python. And these are images that my colleague had created. So efficiency of analysis. One of the things that people can get really stuck on is sometimes it just takes a while for one of the algorithms to run, right? There's ways that we can optimize that, and there's ways that we can allocate how we're spending our time. One of those things is using a template. If I start from a template and I drop my data in, I adjust the gates, and then I can focus just on optimizing that high dimensional piece, my TISNIs, my UMAPs. I can spend the time where I want to, so I'm getting the analysis back that I want. I also have that ability to test multiple different formats, right? I don't have to just be like, oh, that was done, and then wait, and then wait. I can just run another pipeline. I'll add, copy and paste, add that second pipeline, change the features just a little bit, see how the new results relate. Is it better? Is it worse? Gives me some ideas on how I'm going to start manipulating this. And then also algorithms are optimized. We do a lot of work in the background trying to make them run quicker, trying to figure out where the sticking points are. Clustering is usually it, right? That's usually the rate limiting straight step. So we want this to utilize all the features of your computer. So I get asked all the time, what do I need on my computer? Guys, more is better. If you have more memory, if you have more cores, we can optimize using all of those so that we can return these results faster to you. Also, like little tools like downsampling, you can downsample and then have your algorithms run a little more quickly. So understanding the whole picture from all of that, all the work that we did. Unfortunately, there is not a one-size-fit-all high-dimensional analysis strategy. I can't tell you like, hey, thanks for calling. I've got this all figured out. because I'd be lying and you would be like, oh, I do not like her at all. There is just a little bit of testing that comes with the territory. You kind of have to take your time, be patient. And I'd like to say that for my beginners, I know it can feel a little intimidating. They're like, oh God, this is going to go horrible. That is okay. If you bomb something, you learn a whole lot really quickly. I have a whole list of never agains. I will not do that ever again. And I know, because I know what it felt like for that not to go right, right? But in this space, while you're working this out, having that beginner mind, you're more creative. You're not restricted to all the things that you think should happen. And you have this ability to just kind of be like, maybe I should just try that. And guess what? Sometimes that works. And then around you, you also have a bunch of experts. That wisdom is invaluable, right? That experience of knowing why this won't work, when it won't work, what's happening in the background is one of the most invaluable tools. So I invite you guys, while you're here, to talk to each other, get to know somebody. I'm telling you, just having those sidebar conversations can really elevate your level of analysis, because you'll be like, I have never thought of that. I would have never thought of that. I would have been sitting there for 100 years and that would have been the last thought to cross my mind. So use your resources, use people that you know, use people that you don't know, get to know new faces, reach out to people that are application specialists, explore different softwares, right? You don't know what tool might be best for you. And I just invite you to really embrace this experience, because we're a very niche group of people, and we're a pretty awesome group of people, if you ask me. So I think that we should just kind of work together and make sure that we're meeting those analysis needs. So if you guys have any questions for me, I'm here to answer them now.
Robin, it's a comment on the flexibility of your platform, because we have interacted a lot. You have helped me a lot with data analysis. And as a situation that happened last year, I had to analyze over 100 titrations. And this was 12 time points per titration, multiple markers, multiple instruments, not only the Aurora. So it was going to be very time consuming, just calculating the stain index out of raw data. So I reached out to FCS Express, and just towards the point that it's not only about unsupervised analysis, and just simple things can be done so much efficiently. And this is going to answer a question. That's why I'm saying this, answer a question from before that I couldn't reply to. And the question was, when you do the titration, how do you know how much antibody you need to add at the end? What's the optimal concentration? So I reached out to FCS Express, and they helped me build this template that I just put the files in, and it's gonna do all the calculation or the linear regression to find the saturation phase, and then define the optimal titer based on kind of that saturation, like inflection point, and do calculation for multiple ways to do the titration analysis, stain index, separation index, signal to noise ratio. So it was great that with 3 clicks for every data set, I can get the final result, which is microliters that I need to add, or nanograms per test. So yeah, definitely it's for me, it was fantastic to have this software available, to just something that it was not available before we talked, but then you guys were flexible enough to create a template. And from there, it simplified the work so much. So thank you, Robin. And just to let everyone know.
Thank you so much for that. And those titration templates are available for us online. So you do have a 7-point, 6-point, and 5-point titration. So if you're working on that side, because that's, again, garbage in, garbage out, good data in, good results out. So if you need to, we have some templates that are already available, and you could just use that as a starting point as well. If you're not sure where to start with a template. You can subtract whatever you need to. You can add whatever you need to. Make it easier, because what you guys do in real time in research, developing these research assays, running them on the instrument, optimizing settings, all of the background stuff is a lot of work. You shouldn't get to the analysis part and be like, oh God, here we go again, right? Like a little reprieve, right? You drop your data in, move some gates around, and then really focus on what are the things that you need your software to do.
So it says, did you also identify the CD39 positive population that was mentioned in Nico's talk? I don't know if you got to in the OMIP, I believe that.
So I probably didn't. I think he found that population in the middle of the night, actually. And I was sleeping because, I have a rare opportunity to sleep. So I didn't see that population, but again, if I would optimize this and spend a little time figuring out what it is that I'm looking for, what I want to see, you will have the tools there that you can do that kind of exploration. And again, you save the template, you save all the pipelines, you save all the work that you've done so that if you need to go to sleep, you go to sleep and then you wake up in the morning and you answer the question about Nico's CD39. So I'm going to have to talk to Nico about this after this. So, but. Or anything you want to see again that you didn't get to see. I'm here in the software. It's your time to shine, guys.
All right, so what if we don't have any questions? Robin will be here to answer other questions throughout the conference. So feel free. Otherwise, what we'll do is we do have one more coffee break before our last speaker of the day. So everyone can go and grab some coffee, talk with some vendors or with each other. This is one of the first opportunities we have to network again as flow cytometrists. So let's hope we can take advantage of that. And thank you so much, Robin.
Thank you so much.
All right, for our online people, as you're still listening, we are going to take a break and we'll be back in 45 minutes.
This the HDMI cable. Just
It's on. Okay. Hello, everyone. Welcome back. And so we're gonna start our last talk of the day. And so Geoff Kraker, he's a senior application scientist at OMIC with 14 years of experience in cytometry, spanning from academic flow course to instrument manufacturers and several cloud-based analysis platforms. He's a persistent advocate for data quality and is passionate about educating people in the field on approachable strategies for high-dimensional cytometry analysis. And it's my great pleasure to have him here. And he gave me a little bit preview on his talk. It's really exciting. So you'll definitely enjoy it. So Geoff.
awesome. Thank you. Is this all right? Yeah. All right. I'm projecting to the microphone. All right. Thank you very much for the introduction. Yeah, I will endeavor to do my best to keep this exciting. I know this is the third presentation you've seen on this same data set. It's a great data set, so I think it deserves 3. But I'll, like I said, I'll do my best to make it worth sticking around for. Some of you, I'm sure, know what OMIC is. Others might not. It was my oversight. I actually didn't put it in the talk title. so that one's on me. Essentially, I'll give you a little summary of what OMIC is, and then we'll talk about kind of how we arrived at the analysis. So actually, it turns out that I'm the one who did the advanced analysis in that paper. So I used OMIC to generate that figure. That was me. I'll talk about how I arrived at that figure that's present in that in the OMIP 69 paper, and then I'll talk about kind of some new, cool, or more efficient ways you can analyze that data using OMIC. So I'll forego the big slide with all the dot plots. I'm sure you've seen that a lot, but basically the problem is still here, right? We have a lot of dimensions. And it can be tough to visualize those. And I like to simplify this analogy a little bit and say, you know, it's like a globe. A globe is a 3-dimensional object, and you can't see all the surfaces at once, but people have developed ways to figure that out. So essentially, they go through and take a globe and reduce its dimensionality, go from 3 to 2. And you can do that a lot of different ways. You can do that in really classic ways. You know, it's in a, up on your classroom wall. But there's limitations here. You know, for instance, a lot of maps don't have Russia and Alaska touching. In reality, they almost, they're almost right there. And there's actually not that many of these maps that show this. This, I think, is, well, and this one too. These two are basically the only 2 maps that show that. But you're making sacrifices when you go from many dimensions down to 2. And the same happens with your data. And so the goal is to be able to use some of these tools to give yourself and then others you're communicating your results to some insight into kind of what's going on in your data. And there's a lot of ways you can do that. So again, quick background on OMIC. OMIC is a cloud-based cytometry and other data type analysis platform. What that means is you access it with your browser. It's essentially just a website you go to, app.omic.ai, and you can set up a demo account. I'd recommend that, of course. Give it a shot. But once you do, you upload your data and all the analysis happens on the cloud. So you can do it from a Mac or a PC, Firefox, Chrome, Safari, Edge, all of those browsers work. And you could even do it from your phone. You know, as long as you have an internet connection, your analysis happens on our server and not on your computer. So you don't have to have a fast one in order to do efficient and quick analyses. This is something I'm seeing more and more, that analytics kind of just expands to take the time that you give it. You have a very large experiment, and you might have some low-hanging fruit. You're like, you know what? I'm going to do a 35-color panel. It's going to be great. I'm going to be looking for these things, these differences between my groups or whatever you're interested in. And on one hand, that's great, but there's also a lot of undiscovered stuff that pops up in that 35-parameter space. And you just might not have the time to sit there and split hairs and figure out what's going on. you get your result and you move on. But there's a lot there still. So you can spend a lot more time on these data sets. And so the more efficient you can be when you analyze these data sets, the more you'll have to talk about. And so we see kind of two main patterns of analysis when people do stuff like this. One is kind of a data science genomics style analysis using programming languages and putting together pipelines. The other is a more interactive, get your hands dirty, backgate a bunch and try a bunch of different algorithms and histograms and 2-parameter dot plots and that one 3D thing you tried to show people, but it didn't really make sense. There's a lot of this that happens in cytometry. And so while we want to enable this interactive, get your hands dirty style, because that's how cytometrists work, including me, we also want to help kind of push the field a little bit more towards a consistent, reproducible, workflow-driven style of analysis, so people can understand, because it doesn't happen anymore where it's just gating and dot plots and then export stats. As people step into this higher parameter space, there's a lot more that people are doing to their data. And so to have a consistent record and a clear record of what you've done, I think is really important. So this is how OMIC is set up. So essentially, OMIC works by taking your data and then applying tasks to it. So one, for instance, is compensation. It's one of the first ones we often do in fluorescence cytometry. And then downstream from that, we only, basically the rest of the tasks only see compensated data. Below scaling, the rest of the tasks only see the transformed data. Below gating, you have your manually defined gates that you can use in the figures or in the algorithms, et cetera. So it follows this kind of hierarchy, and it makes it really obvious kind of what order you did things in, and then what settings you used for those algorithms. So you can do all the basic things that you need to do in cytometry in OMIC. You can edit compensation matrices, you can make new ones, you can, again, change the scales, the mins, the maxes, and the scaling transformations, build figures, export stats, export your FCS files if you want to. So you can do all the basic stuff. We have some specialized plot types, especially as you're getting into higher parameter space. It's always good to check to see if you're in mixing or if your compensation was appropriate. And so we have a really quick, easy way to make n-by-n plots. And then we have these kind of special x-axis plots. So you can either use these for functional markers, like here we're looking at PD1. You can use them for like titrations, where each one of these essentially populations is a different file. And then we'll see a version and a little bit in the presentation. You can change that to have each one of those different columns be channels as well, to, I think, in my opinion, great effect, but I'll let you be the judge. Another big part of OMIC is sample-based metadata annotations. So you tag the samples with your experimental group, so how they were treated in your experiment. So what really makes sense are things like different time points, or different sample types, or different treatments, things like that. Also is really helpful is if you have different dates or different batches. If you have a longitudinal experiment, it's really important for you to be able to know which files belong to which batches. And then you can look for batch effects really, really quickly using these annotations. These come into play in gating. You can use them in figures. You can use them in algorithms or in our differential analysis tools. There's a lot of parts of the software that can use these, and it can really help you kind of split apart and look for differences between your groups. Another thing that I really wanted to talk about just briefly, and we'll see it once we hop into the software, is virtual concatenation. So in OMIC, because we have all the data up on the server, you can essentially stack whatever files you want to in whatever plot or whatever algorithm you want to, whenever you want to do it. You don't actually have to concatenate the samples physically, I mean digitally, I guess. We take care of that for you. So here we're concatenating, again, with some of that metadata. And we can, again, hop in the figure and actually see how this works live. But I wanted to take a second. So before you run algorithms, you don't have to concatenate your data. We do that in the background, and then we split it back out into individual files for you. And then also, when you're building figures, you can use that metadata to really quickly concatenate these labeled plots and flip through and look for differences. So here you can see, for instance, this population is very abundant for the female samples here, and very less abundant or sparse for the male samples here. And this is a qualitative way to kind of investigate these. And there's more quantitative ways we can use as well. On top of the basics for flow, we do dimensionality reduction algorithms. We have a bunch we can pick from, and I'll show you what those look like. We have clustering and heat maps. We have differential stats. So in this instance, this is an algorithm called EdgeR that looks for differences between the abundance of two groups in your data. But if it spits out a significant result, you say, oh, all right, great. Cluster 12 as a big fold change and is significant. Fold change or magnitude is on the bottom, the x-axis, and significance is on the y-axis. You think, great, Geoff, that's a green dot. But what does that mean? Is it just a single sample driving that significance? Is an outlier? What's going on? And so you can then take this information after you've clustered, you've done these differential analyses, and you can look at kind of what's driving, what the distribution of your samples are between your experimental groups. Again, using that metadata that we've concatenated or that we've annotated these with. We also have some data cleaning algorithms, especially if you're looking at lots of samples or you're not the one that acquired them. We're getting more collaborative in, especially with these big experiments. So if you're using a data that you aren't as familiar with, you can run it through a data cleaning algorithm like Peacock or FlowCut. It'll trim out the clogged events, or if there's signal change over time, it'll pull those out for you. We have normalization algorithms. I have a whole talk about, and I won't do it now, but about why you shouldn't normalize and you should just prevent it from happening in the first place. But it turns out nobody comes to talk to me beforehand. They always have batch effects. They want to talk to me afterwards. They say, how can I fix it? So there's a lot of caveats involved, but there is certainly some solutions sometimes that you can use. And just a few more things before we actually hop into the fun part of the presentation. Our emphasis is on performance. We have people with over 1,000 files in a single experiment. We have people using over 1 billion events in a single experiment, and it's built to handle things at that scale. The algorithms themselves are not designed to handle that many events, so you have to be a little bit more selective about the comparisons you're doing. But it's not necessarily a bad thing to have all of the events or all of the samples from your longitudinal study all in one place. Then you can be picky about kind of what comparisons you're doing. We do have a more regulated version that we can turn on for people doing more regulated work. I can happily answer those questions later afterwards. We have a way to export the data with batch reports as PowerPoints or as PDFs. You can export the new channels that you've added to these FCS files with clustering or dimensionality reduction. You can export those back into FCS files you can download. And you can also export stats as a CSV. That's pretty standard behavior for cytometry software. Then again, something I'll just mention here quick and then move on, but we also have auto gating. So if you have a consistent panel you're going to be using on a bunch of samples and you want to build an auto gating script for that, there's a group here at Dotmatics that does that. So again, happy to answer questions after about that. So how did we end up with that figure? I'm sure you've read the paper, or if not, you should. But basically, there's a whole series of manual gates and the gating hierarchy associated with that. And then the second half of the figure is this. Essentially, it's a UMAP with different colors, different clusters on it. And so, how did we get there? I mean me, I got there by keeping in mind this workflow. This is there's a lot of different branching you can do. This is just a broad summary. But basically, the data was acquired. It was unmixed, and this it looks simple. It's just one bubble. No, this was like most of the paper. It was an incredibly huge effort by the team at SciTech to get such clean data. If you haven't played with the data, I'd really recommend it because it's so fun. It's 40 colors and it's so clean, and you can find all these fun little phenotypes. So maybe that's just me. Maybe I'm the only person who would do that. But I really enjoyed this talk prompt because we got to revisit it. So the data was unmixed, and then from there, it's kind of up to you how you want to tackle this. And in this case, I checked to make sure the data was clean. I did UMAP as my dimensionality reduction step. Again, not necessary, but I think very useful. I think more people could run dimensionality reduction on most of their experiments and be better off for it. Then ran FlowSum to split those cells into different clusters. And then this particular data set just had 4 samples. So there was not a lot of statistical inference you could do. There was no kind of experiment behind it other than to assemble the panels. So there was an incredible amount of work done, but it wasn't actually necessarily asking a scientific question like that. So I didn't do any of that. But qualitative visualization, there's lots of those. And so you'll see in the figure, again, like I'll mention, that slide comes up in just a second here. And then a lot of the work that I've done, for this particular talk, on top of that, there's some fun ways to kind of split up and look at these populations. So I wanted to take a second, and for those of you who haven't tried using these algorithms, and you're like, yeah, that seems like a stretch. I only do like 10 color stuff, and it's not something I really want to try, or I don't have the opportunity to. Number one, don't be intimidated. You can certainly use them in R, and if you're feeling scrappy and like you want to learn a programming language, I'd recommend it for sure. But a lot of people don't have time to do that, or motivation. You're like, look, that's just a whole thing that I'm not trying to get into right now. That's okay. I feel that way too, and that's why we have this software. So there's a lot of software out there. A lot of people have access, like you've seen over the past couple talks, to software that can do these things. But I'll talk just really briefly. I promise I won't get too into the weeds on these settings, but just basically about why I made the choices I made. So first up, when you set up these algorithms in OMIC, again, you don't have to concatenate ahead of time. Basically, you just choose which files you want to include as part of the run, and then you can move on from there. So you can exclude stuff if you want to. You select which features you want to include. And basically, I'd recommend, if you're going to try this yourself, and for this, using all of the markers you would use to gate. Anything you want to use to phenotype things, you can include them. Some people have questions like, hey, can I include functional markers? What about things like CD4 that kind of respond to stimulation but are mostly a phenotyping marker? Great, include that too. If you're talking about things that are like exhaustion markers or phosphosignaling markers, you can include those in this list as well. But if they change with stimulation, you'll get a weird little peninsula bumping out of your island with those activated cells in it. And that's okay if that's how you want to report your data. So there's no right way to run most of these algorithms. There's a ton of correct ways you can run them, as long as you're comfortable explaining to your audience, whether it's the granting committee or your PI or whoever, the lab meeting with that really mean other lab PI who always makes you feel bad. So if you can explain stuff like rock and roll, do whatever you want. But there are some things you should not do. Number one, never use time in these algorithms because that doesn't make sense. It's cell extrinsic. It's just when the event happened. So that doesn't make any sense. And a lot of the time, I tell people to avoid scatter because if you're not transforming these, basically the values for these channels will be very, very, very large, and they'll swamp the contributions from all your fluorescent channels. So I recommend people don't include scatter unless you're doing like microparticle work and you are making them arcsine H scaled or something. And then, yeah, absolutely go for it. Another thing really quickly, if you've already cleaned up the data, so for instance, if you've already gated on just your live cells or just your 45 positives, if all of your events are relatively homogeneous in a particular channel, you don't need to include those. They're not going to help pull apart your populations. If you're looking at bone marrow, however, you might want to keep 45 in because there's going to be heterogeneity there. Or if you're looking at tissue, or if whatever. So definitely feel free to tinker, but time is definitely a no-no, and often not scatter parameters. But you can clean up and gate on your scatter parameters and use that gated population downstream. Then when it comes to these settings, these are not the ones that I used in the article. If you go to the supplementary information for the OMIP 69, there's a whole description, and I think it's in materials and methods too, about the settings I used for the algorithms in there. So I'm not going to dive too deeply into that, but in this particular case, if you want to try it yourself, the defaults are pretty good that we have in our software. But by all means, I think a lot of people could be better served if they experimented a little bit with different settings and found which hyperparameters are the best for their particular data set. It'll depend on how you've designed your panel, how well you stained the cells, how well you've titrated your antibodies, what the heterogeneity is of your sample. If you ran your panel on single cell from a culture or PBMCs or bone marrow, it's going to behave differently and you'll require different settings. So definitely feel free to tinker with these. In fact, like I said, I'd encourage it. neighbors right here, basically, it's a good default. Starting with 15, 30 is okay. I don't go much above 60. Again, I promise not to get too into the deep weeds. So if you have questions about your particular data, I'm happy to take a look at it after or the next couple days. Minimum distance is what controls basically how far apart your islands are in UMAP. And so if you want to really emphasize the separation between your cell types and really separate them, get a lot of white space in between those islands, you can lower this minimum distance to something like 0.1, 0.05, things like that. If you want to get a lot of insight into the heterogeneity inside of an island, like the differences between your naive and your central memory T cells, you could increase this minimum distance. And the technical terms for these things are local structure for that small differences inside of your islands, and global structure for the differences between your large populations. And the rest of these I'll just leave alone. But the eagle-eyed among you may see something that's a little bit weird, and I'll come back and talk about that at the end of the presentation. The next algorithm I used was FlowSum, and this is for clustering. Basically, clustering just puts your cells in a certain number of bins. You say, hey, I've got maybe about 30 phenotypes here that I'm interested in describing. So I might tell it to make 40 different clusters. And that way, if I have weird debris that I missed, or if there's weird little like dead cell populations or things I can't explain, those can eat up some of those extra ones. So I get the things that I care about in those 30 clusters. The same holds true here. Same number or same way to select the files, the same number of features you want to run on. And we'll talk about this in a little bit as well. And then FlowSum settings here, I'd recommend running these a little bit higher. The default settings for FlowSum, I think for a lot of people, is 100 clusters, and then it takes those and kind of tries to combine the similar ones for you. I would go a little bit higher. I normally do like 200 or 300 clusters, and then have it combine that. And then the real question I get probably most frequently is, what's the right number of clusters? This is something that doesn't just plague cytometry. This is like a pan data science thing. Like, how do you know when you've got the real correct number of clusters? And I take a very kind of pragmatic cytometrist view on this. So this is just my take. It's not the right way. It's just the way I do it. I say, can you find your smallest population of interest in one cluster? And if the answer is yes, then great. That's the right number of clusters. If the answer is no, if it got sucked into some other cluster and you can't separate it, or if it split your population of interest into 18 tiny clusters, you either have not enough or you have way too many. So this also requires some iteration. You have to sit down and say, all right, maybe I want to run FlowSum with 10 meta clusters, and then with 40, and then with 60, and then see which one gets you your data in the right number of clusters. And if you're doing discovery work, you're like, look, man, I don't have the smallest population of interest. I want all of the small populations. This is the discovery work I'm doing. I'm trying to see like total what the differences are between healthy and lupus or something. Like I want this unsupervised analysis to tackle that question for me. Then you can just skew high. You could do 400 clusters and condense down to 200 if you wanted to. So again, there's not a single correct answer. And people have tried. There are algorithms out there that try to essentially kind of take a step back and say, I'm not going to request a hard number from you, but you need to tell me kind of what the size of the neighborhood is that I should be looking at. So things like Phenograph, they don't have a cluster number. They don't have a K or a meta cluster number, but they still have a setting that you can change that will change the number of clusters that Phenograph spits out. And the same is true with Park, which we'll see in just a second, and a handful of other algorithms. So there's not an easy fix yet. People are working on it, and once they figure it out, they'll make a lot of money. So yeah, keep your eyes peeled. But in the meantime, it requires some biology knowledge as well as some experience or some investigation. All right. So how did we end up with this figure? This is certainly, I mean, I kind of like it, but that's not a surprise. But how did we end up with this? So this is, like I said, UMAP and then clustering after UMAP. And so another question I get frequently is like, does the order matter? Like what's going on with these? And the answer is, there's not a single correct way. I do it this way for a reason. and we'll get into this in just a second, but if you want to run your clustering first and then UMAP after, that's absolutely okay as well. The reason I run it this way, and the reason why I have these relatively nice, clean separations between these different groups here, is because I use the UMAP parameters as clustering channels as well. So I use some surface markers and the UMAP parameters in my clustering. This is kind of non-traditional. It's not really a settled thing in the field, and I'll tell you why. But again, this is not the only right way. It's just the way I do it. And so I think it gives me a great kind of balance between having UMAP do the heavy lifting and separating my populations for me, and FlowSum to also kind of add some nuance there and help me get the kind of more homogeneous populations in my clusters. But I did that work. I got those clusters, and then from here, you have just, I mean, if you ever tried it yourself, just kind of a confetti-colored group of clusters all laid on top of each other in the UMAP channels. And so I think it helps interpretation of these results. I think it's a little bit easier to digest for people you're showing it to if you kind of make it cohesive with the coloring. So I'll tell you all kind of how I did this in OMIC as well. It's actually not that difficult. And I have a link to the website I used at the end of this presentation. So there's a little resources slide. I don't know, are we giving these presentations? Are we posting them publicly? Yeah? All right, cool. So you can go check it out so you don't have to furiously like scribble down a link or something. But yeah, so I would recommend, even after you've done kind of some of the heavy lifting computational work, you can still, I think, get benefits from polishing it a little bit and making it easier for your audience to kind of understand what you're going for. So where can we go from here? Actually, before we do this, I'm going to hop into the software and kind of show you what I did to get to that point, to get to the cluster point. And then we'll talk about the new cool stuff that I've kind of got prepped to talk about. All right. Is this coming through? Yeah, perfect. Okay. So this is what OMIC looks like. Again, if you want to try it out yourself, you can just go to app.omic.ai and sign up for a free trial account. But once you have your data uploaded and you've clicked on that data set, you can see a quick summary of all of your files that you have. Again, here it's just a little over 2.5 million for these 4 files. You can tag them with dataset notes. So for instance, if I wanted to tag this as, coming from SciTech, I could do that. And maybe, let's see, panel, I could make it an OMIP panel if I wanted to. And then now this is tagged and I can search for this data set with these values if I wanted to. Doesn't matter that much if you've got like 5 experiments, but if you've got like 500, it comes in handy. The next tab over is file metadata. Again, there's not a lot of really crazy groups to dive into here. It's not intended to be that kind of experiment. But I'll show you what it looks like on a different data set. So this is another data set from from Flow repository. And it is a little bit more interesting. It's looking at human PBMCs with COVID infection and comparing male and female responses to COVID infection, and also looking at some different time points. And so you can see here for each of the files, we give it a little ID number, and then you can pull in any keywords from the FCS file itself if you annotate those at the cytometer. You can add in any of these annotations manually that you want to. And then maybe you are looking back at data, some like retrospective study, or you get like outcomes data from a clinical trial, and you're like, oh, now I've got these particular patients responding to therapy, and these ones are non-responders, or I've got survival, or I've got severity of disease. You can do a whole analysis and then come back, add that information in, and then use those tags in further figures that you build. So it's not like a static thing. You can add to it. You can change the channel names, the target names here if you want to, or the configuration names. If you've got two different instruments and the service engineer labeled one FTSE and the other Alexa 48, and it's just the first blue channel and you want to make sure they line up, you can fix that here. You can collaborate very easily with other people if you want to, but when you upload your data to OMIC by default, it's just private to you. Nobody else can see it. And then the workflows here, this is where you can actually start analyzing the data, building your workflows, and you can have different strategies. You can see here I have a bunch of different workflows for this particular data set. They all reference the same FCS files, but they're different strategies. Some are just manual gating. Some are all unsupervised algorithm driven. Some might be from other people who have access to this experiment. So there's a lot of different ways you can tinker with this to get a decent analysis done. And then you can save these workflows as templates if you want to, or share just the workflows. It's a very flexible system. So we'll go back to this particular OMIP 69 data set, like I said. This is that workflow that I've put together. So if I was going to make one from scratch, which I won't, I won't build one from scratch here for you all, but I'll show you all how it works. We have this whole list of tasks. So when you first upload fluorescent data, if it has a compensation matrix in it, whether it's spillover corrected or compensated, that first comes here in the compensation tab, similar to like what we saw on the slide. Next up is scaling. To make sure you have the min and the max adjusted correctly, make sure your ArcSine H scaling is adjusted correctly. We use ArcSine H. Other people have added this recently. Bi-exponential is a really classic way to transform your flow data. We have some reasons for why we prefer ArcSine H, but you can adjust that information here, and you can see how things are scaled this way. So we have default set per cytometer, but you can change those values if you want to. If you prefer, a certain amount of white space below your negative population. I know some people are very particular about this. You can adjust the min and the max that way as well. So the next, kind of step in the process is, in this case, gating. Although you might do like normalization, or you might do one of those like flow cut or peacock cleaning tasks. You can add again any of these tasks to your analysis if it fits your particular workflow or your assay. So I think of OMIC as like a kind of a large toolbox, and you get to combine these tools together to run an analysis. So it could be very simple, or it could be a really, really complex analysis. And the rest of what we're going to see today skews towards complex. So sorry if this is intimidating. You can really do a lot of simple stuff quite easily and quite effectively as well. So this is the gating interface here. So this is all of the populations that I've drawn to kind of match the first part of the figure in the OMIP 69 paper. So you can see there's a lot going on here. I've done things a little bit differently in that I've excluded populations after identifying them. So in this case, I have 123 positive, and then from then on, those are gone. So I identify like basophils and PDCs, and then all the 123 negatives I carry forward, and I start kind of pairing off populations that way. And I found that gives me kind of a pretty accurate count of my cells kind of on the back end. But in OMIC, you can do, like I said, this is a kind of a normal gating interface. You can also use Boolean filters if you want to, and or not. And if you're doing, for instance, like polyfunctional T cell stuff, and you want like every permutation of your activation marker or your cytokines, you can make combo gates really, really quickly. You say, hey, take these 4 gates and give me every positive, 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 negative, et cetera. You can kind of have it auto make those for you. In this case, I spent a lot of time gating all this, and then we have these as reference populations. So again, kind of in an effort to go one-to-one with the paper. But if you want, you can see kind of how each of these are gated for each of these particular populations. And I'm not going to have you all sit here and watch me do this from scratch. You can trust, you can trust that I did this decently well the first time. But I'll show you how to check the gating hierarchy so you can actually see if I did things decently. Downstream from gating for phenotypes, I subsampled on just a clean population. And then from here on, everything below this particular subsampling task is using all of my events, but only the clean ones. So I'm getting rid of all the doublets, the debris, the dead cells, etc. So you can subsample for specific populations. You can subsample or basically exclude files. If you upload, for instance, FMOs to set your gates, but you don't want to include the FMOs in your advanced algorithm analysis, you can basically remove those files. You can actually remove channels too. So if you have empty channels or things like the side scatter blue, if you're using an Aurora and you don't, I mean, you should, because it's kind of cool to pull out red blood cells, but if you don't use it for that and you don't want it to appear downstream, you can remove any particular offending channels if you want to from the analysis. So after this subsampling step, essentially kind of the sky's the limit. You get to combine whatever tools, whatever algorithms you want to analyze your particular data set. So in this case, I have a lot here, and I'll show you what I did to do like that normal, the figure that we looked at, the figure that's in the paper. So here I did UMAP, again, on all of the 4 files. I did all of my surface markers, including some of like the dimmer ones, or the ones that like, are a little bit more kind of functional markers like 57 or PD1. So I included all of those. And then I have my UMAP settings kind of set up to give me a pretty reasonable look at kind of what's in this particular analysis. And then I got a UMAP that kind of looks like this. So it did a pretty good job of separating out all of my different populations. And then after UMAP, I ran FlowSum. And if you're like, wait a second, he's not doing this live. What? Running things on 2 million cells doesn't happen in minutes. And so in the interest of kind of walking you through stuff, I did this one a little bit faster. So in this case, this is on 600,000 events, and it got done in, a little under half an hour. However, if you wanted to look at how the 2 million or the 2.5 million event one worked, we can take a look at that in just a second. So that's UMAP. If you're curious, I mentioned, like I said, playing around with these. If you're curious with how these might work, or how you might tinker or iterate on those settings, once you start one analysis in OMIC, you can duplicate it and then change a setting. So for instance, we might change this to 60, and then I'll click run. And then now this is off and running on our server. So I could, close this browser window and it was still complete. Send me an email when it's done. And I can duplicate this and open this, and I could change this to maybe like 0.2 and click run. And you can repeat that as much as you need to. So I could have 20 of these running concurrently if I wanted to, if that was going to be useful for me to figure out what different hyperparameters or settings I should be using for this particular data set. Turns out, number one, it's not necessary because I've already done that. But if it was a new data set, then yeah, absolutely. You can do that. And I like to think of running these. It's not like p-value hacking or something like that. Basically, it's like, and I was explaining to somebody, I was explaining it this way yesterday. It's what you're doing is trying to get the best kind of lower dimensional representation of your many dimensional data. And by changing these hyperparameters, you're changing kind of how you're rotating it, basically. It's a little bit of a conceptual leap, but functionally, I think it makes the analogy work out. So if you were taking your hand and shining a flashlight on it, you're going from a 3-dimensional object to the shadow on the wall, that'd be a 2-dimensional object. Well, kind of 2-dimensional, but picture. And if I shone the light this way on my hand, it would look like a fin or something, and so it's not the best 2-dimensional representation of a hand. But if I showed the light this way on my hand, everybody would say, oh yeah, that's a hand, so that's a better representation of my many dimensional object in just 2 dimensions. And that's essentially what we're trying to do here. When you're changing these hyperparameters, you're trying to get more insight or a clearer insight into the relationships between your different populations. So moral of the story here is go nuts. Really try to figure out what the best settings are for your data in your panel. All right. So after UMAP, after we ran UMAP, I ran FlowSum. And again, kind of some selected surface markers. This is just it's the way I tackle it. It's not the way you necessarily have to. If you do it this way and you show some computational people, they might be like, why are you doing it that way? And you can answer, well, Geoff Kraker told me to. And that was, and that's technically, I didn't tell you to. No, honestly, there's, like I said, there's some unsettled debate as to if it's effective or not, or if you're losing information. If you're only feeding it the UMAP parameters, you're giving up that Russia-Alaska connection, like I was talking about. And so that's why I also put in some of these kind of hard to nail down or smeary interactions that are important for me, the biologist, making these phenotype decisions. So things like RA and CCR7 or 127 and 25, or the things that you really want to make sure you have separated well, I add those markers in. Like I said, I usually use more than the default number of clusters, and then these get combined into, in this case, just this number of metaclusters, and that's basically the process I used for that figure. So if we wanted to see what this looked like, we can then, after FlowSum, and again, in full transparency to see how long this took, this FlowSum, I don't know if you can see these numbers, was started at 3:30 and ended, sorry, 3:39 and ended at 3:42. And so FlowSum built to be really quick. It's a really snappy algorithm. But now FlowSum got us some clusters, and then we can use these. We can turn them into populations with the gating task. Under here, categorical filters, we can kind of do this automatically using OMIC. So we can choose our meta-clustering channel, and I'll give it like a little prefix so I know kind of what populations I'm working with here. And then OMIC kind of auto gates around these populations. And now I can click save. And we can make a figure to kind of see what's going on here. So as I'm kind of clicking down through these tasks, you'll notice I'm kind of building these hierarchies. So downstream from this FlowSum task are those FlowSum clusters that we just used. But those FlowSum clusters aren't present below these UMAPs because that FlowSum hasn't been run. So it very strongly follows this hierarchy. So downstream from this gating task now, I'll make a figure and make a new plot here and make an overlay plot. And you can see here, this is where I can start concatenating my files together if I wanted to. Actually, we'll change this right back here just so you can kind of quickly see what's going on. So I can really quickly, like I said, start concatenating virtually. So here's one file together, but here's what it looks like when I'm stacking these two, 3 and 4. Again, this is really, it's quite simple to do this if it's necessary. So I'm going to make this overlay plot and concatenate all my files and then color by FlowSum cluster. So any of the pull down menus that you see here in OMIC, if they have this little like blinking cursor search function, there's a lot of populations here, all my manual ones and also my FlowSum clusters. I can just search FlowSum or FSum, whatever I titled that, and then select all, and then it builds that basically that figure right away, and I can see now where each of my clusters are. So this is again not colored well. It takes time to do that, and like I mentioned, I'll show you how I did that right near the end of the presentation. But this is effectively the analysis that I did for that paper. So this is where we kind of left it. The paper wasn't intended to kind of describe new biology. It was mostly a technical treatise, an opus. It was a huge manual. If you haven't read the supplementary information, that's where like the bulk of the work of the paper went. It's a great resource. And a lot of people's Aurora data that I see now, if they're tackling something around 40 parameters, use most of the same antibodies and most of the same colors just because so much work went into making it so clean. I think that speaks volumes to how much work went into it. But that's kind of where we left it, was getting FlowSum clusters on a UMAP and calling it good and saying, hey, this is where all the populations are. So I'm not sure if anybody has noticed this, but there are some gray colored populations here that conspicuously don't appear labeled in this figure. And I'm gonna come out and say it, like, obviously, number one, that's on purpose. This one in particular was pretty vexing to me. It looked just like monocytes in scatter, and in every other parameter, it looked just like a classical monocyte, except it didn't have CD14, just no CD14. And for me, I'm not a classically trained immunologist. I had no idea what that meant. And so I said, well, we're just not going to label it and hope nobody asks about it. So here I am outing myself in front of all of you. That was totally on me. So if anybody knows what that population is, find me after and tell me, because it's going to be like haunting me for the rest of my career. I'm like, I don't know. I wish I had been able to describe that. But this is essentially what happens when you're looking at really high parameter data. Sometimes there's stuff that pops up, like in Nico's talk, that only 39 positive. You know, like what is that? I don't know. That's stuff that you find when you're doing these really, really broad, in-depth examinations. So that's how we ended up with that. Now, where can we go from there? And number one, I'm going to talk about kind of how I labeled these, because this is if you've ever tried to do this, a huge thorn in your side is going from FlowSom clusters 1 through 50 to all of these named populations, right? This is kind of like a goal of the field. This is really low-hanging fruit of like, how do I label these things? Because it is arduous for sure. For this figure, I spent like 3 days and like 2 whiteboards and like scribbling like cluster number 37 and 42 are this, and I'm gonna make a little OR gate and kind of glue them together, and I'm gonna relabel it. And it was this huge manual effort, and it was compounded by the fact that I like actually did 120 meta clusters, and so then I had to glue them all back together into stuff that made sense. And so I kind of shot myself in the foot there. But essentially, there's not an easy answer to this out there. There's a lot of people working on it, but there's not an automatic, just like finger snap way that you can all the time count on having these labeled. So there's typically a variable amount of work involved to make sure that you're looking at this. Typically, I recommend to people, if you're going to try this kind of workflow, and then look at differential analysis between like your responders and non-responders. What a lot of people are doing, I think it's a great application of these tools. But if you're going to use this strategy on that, before you spend a day trying to label all of your populations, first check to see if any are significantly different. So it's great if you have, out of 50 different clusters, 3 of them are significantly different between your experimental groups, and you can comment on those and kind of chase that down. Then start finding out what cells those are. But don't get to this point, get really excited, label everything, and then do the statistical analysis and say there's actually no differences between my groups, and I just wasted all my time. So just pro tip, FYI. But how did I do this? So basically, I looked at heat maps of all of my clusters versus all of my channels and said, all right, it looks like cluster number 5 is 4 positive, 3 positive, 8 negative, RA positive, CCR7 positive. And I just kind of like went through and made a little note and then like moved on. Then I checked that against where I would draw a naive CD4 positive T cell gate in RA versus CCR7 or whatever I was using. And I said, yeah, all right, that fits. I would include that. I'd label that as that particular population, and then went from there. That is pretty tedious. And so this next slide is, again, just a little, hopefully something that can improve your life, because this is something I've started doing since that process that has made it a little bit better. And that is to, this is in Omic, so I'll describe it here, but you can do this in whatever your own software that you're using. If you're going to make a heat map, you can choose what populations you're going to be looking at in that heat map. So in this particular case, oops, I'm gonna come over here. When I'm setting up this heat map, I say, hey, look at all of my files, and then look at all of these channels, and then look at the medians of those channels, and then do it across all of my clusters. So here, this is kind of the meat of the heat map. But then I also include my manually gated kind of benchmark or landmark populations. So what happens is these get looked at by the software and included in the heat map as well. And then you have labeled populations getting hierarchically clustered right next to your unidentified clusters of interest. So it gives you a really quick idea. Oh, everything in this branch looks like a naive CD4 T cell. And so what does that look like? So it looks like this. So now what we're looking at here is every surface marker across the top. And then along the side here, each row is a different cluster or a different manually gated population. So down here, if I look at these, this is the phenotype of my CD4 positive, 25 positive, kind of like Treg-like cells. And they're 3 positive, 4 positive, 8 negative. They're 25 positive. I mean, 25 is kind of a dim marker, so it's not screaming bright, but it's 25 positive, 127 negative. And then right underneath it, right here, is this cluster number 24. I haven't labeled this yet, but this has the exact same phenotype, 1 for 1, between these two. So much so that they got clustered right next to each other with this little dendrogram. So I know right away, I don't have to jump through any hoops. I don't have to like recite the phenotype in my head. I can say, boom, all right, I know I can now relabel this one as my clustered Treg population. So number one, this is like a shortcut to figuring out your heat map. Seed it with already predefined manually gated populations, especially in something like this. If you can get to them just super quick, like doop, doop, doop. Even it doesn't have to be like RA positive Tregs or, really hair splitting populations. But even putting things in the right neighborhood, like naive CD4s, naive CD8s, NK cells or whatever, it can really help speed up your labeling process. So number one. Number two, I talked about these special x-axis plots. I'll tell you how to make figures with these. So we'll do it in just a second. But basically what's going on here is I'm taking my total cells. Again, this is, it's really easy to concatenate kind of automatically in OMIC. I'm taking total cells, or unfiltered, as the background. Then I'm overlaying one of the clusters on top of these. And so basically it gives me context across whatever channels I choose. In this case, it shows kind of some of the big important ones for phenotyping. And I can really quickly say, all right, so what's the phenotype of this? And this is kind of like a heat map. It gives you, this one's positive, and this one's positive, and this one's positive, and that one's negative. But it also gives you distribution and abundance of the cluster information. So it gives you a really snappy way to go through and really quickly in your head kind of figure out what population cluster number one is. And in this case, if we look at it, it's RA positive, CCR7 positive, CD3 positive, 28 kind of dim, and 4 positive. So, pretty classic. That's why there's a lot of cells here. You can do this. I mean, so if there's a rare population, it shows up like this, obviously way less abundant. And these, it turns out, this was a really, really tough population to track down in this data set, but it's CD141 positive dendritic cells. They're definitely there, and they're screaming bright in 141, but it's really easy to lose them if you have kind of too restrictive of a scatter gate, and if you don't kind of know that they're there and need to look for them and make sure you're gating them all appropriately. So this is, again, just an indication that there's actually a fair number here, but it's just a wicked rare population. And so you can see that here, this happens to be in this particular branch of my analysis, cluster number 32, and it shows up really bright there. So I'll hop into the software now and show you how to make these plots, because they can be really useful as you're trying to label these populations. All right. So you have to be beneath, a branch of your analysis that has those populations to work with. And I'll make a new figure now. And I'll do a new plot. And this is an overlay plot kind of at its base. So I need to make a scatter overlay plot. And again, we're going to concatenate things, all of our files and some of our populations. We're going to overlay some populations. And here, I'm going to make unfiltered in the background. And then I'm going to overlay my first cluster of interest. The thing is now we're not looking at necessarily the channels we want to be looking at. So I'm going to take this and I'm going to make it, I don't know, something like CD14. And then scroll down here, there's a special x-axis mode. So if you make it file, this is how you get those kind of nice titration plots to show you how you're staining across multiple samples. We're going to, for this particular one, use features. And what that means, features in OMIC are like their channels. And so now we get to select what channels we like to use in this particular one. So I'm just gonna, I'm not gonna do everything, but I'm gonna highlight some of these main ones and go from there. I don't know, we'll do that and 25, sure. Okay, so we've got some of these, and as you can see, as I add them, they kind of click in here and they start filling in. I'm going to unlock this so I can make the x-axis much larger. Is that too big? No, that's way too big. Yeah, there we go. That'll work. And I'll make the y-axis a little bit bigger as well, just so we can get a little bit more dynamic range there. And so now this is kind of like approximating what I just had in the slides, right? So this is cluster number 1 overlaid on top of total cells across all of our different, well, some of our surface markers. So now in OMIC, you have this really cool feature that is called permute. So when you're building figures in OMIC, you can permute a plot. What that means is make a bunch of new plots based on a rule, and then you get to pick what that rule is. So in this case, I'm going to say permute, and instead of changing the file, make a new plot for each file, or make a new plot for each x-axis, I want to make a new plot for each filter that we have for each of our clusters. So here I'm going to type in fsum to narrow it to just the 50 FlowSum filters that are populations that I did. And then I'm going to select all. So I'm going to say here, make 50 new plots just like this one, but iterate through each of my clusters. And then I'll say, make a new row for each time you do this. And I'll say permute. And so now, that's the first one. And then it goes cluster 2, cluster 3, cluster 4, cluster 5. And if you want, OMIC works in multiple windows. So you could have your cluster labeling task pulled up on this side and this on this side. And you could just go through and just start one by one labeling each of your clusters. And because this gives you so much more information than a heat map, it just makes it a much more direct path to getting that information recorded in your cluster names. So permute works across different metadata tasks as well. If you want to do different batches times different time points, you could do that. It really is a very flexible way to interact and work with your plots and your figures. So definitely take advantage of it. If you don't know if something will work or not when you permute it, I'd say give it a shot. Frankly, I've been floored, even like almost after a year working here. I've been like, oh, that works. That's fantastic. So yeah, give it a shot. It's built to hopefully behave in very intuitive ways. But if you think of other cool stuff, again, we're always happy to take feedback. So if you see this and you're like, what if you could do X? Let us know. We're happy to add stuff to our feature list. I'm not promising when it gets put in the software, but we'd love to hear it. All right, so again, this is just another kind of quick pro tip for as you're trying to label populations. It can be nice. So a little change of pace here, and the kind of the difficulty or the higher concept stuff starts to increase. So I wanted to take the first chunk to talk to the people who hadn't seen OMIC before or had heard about it, or were curious kind of how I developed that particular figure. But from here, I thought, you know what, the next conceptual step is what new cool stuff can you do, or what stuff are people trying? And so this is one of them. So sorry if it seems like I'm going off the rails or it's all of a sudden like a big whoosh thing. But I think this is really cool. And it's experimental, not like totally novel. I'm not the one who developed any of this stuff, but it is kind of cool. So Apologies if you were like barely hanging on, because we're gonna we're gonna take it up just a notch. I promise it's not gonna be too long, but it will be fun. You know, I talked about kind of making your figures easily accessible, or like making them easy to digest for your audience. That's a serious consideration. You know, if your manager or your PI is sitting there and you're like, here's UMAP, and they're like, great. And then you're like, but have you seen TriMap? And they're like, what is that? And you're like, well, let me tell you. And it turns into this whole thing. Like that's added kind of friction in that communication that you don't necessarily need. Stick with UMAP. They understand it. Rock and roll. It's easy. But this is going to be one of those things that will make it harder to explain, but I think it can pay off. So one of the principles that I learned really early on in the flow core is, you know, you can draw gates on histograms. That's totally okay. You can do that. However, if you draw gates on your marker of interest and then add another marker in, you have all these parameters. You can certainly use them. And especially for stuff like this, looking at scatter versus live dead, monocytes typically just suck up a little bit more of the live dead stain. So you don't want to put a hard gate right here necessarily, because you'll probably end up gating out some of your actually honest to goodness live monocytes. And so there's, I think, a benefit to looking at, you know, instead of just one dimension, to looking at 2 dimensions. So conceptual step, what if we looked at more than 2 dimensions in your dimensionality reduction? You can do this. You can do 3-dimensional T-SNE results. But what if you did like an 8-dimensional result? You can do this in UMAP. So you can say, UMAP, give me 8 dimensions instead of just 2 dimensions. Number one, that makes it really hard to explain to an audience kind of what you're trying to tackle. But basically, if you end up using these UMAP dimensionality reduction parameters in your clustering, it gives the clustering algorithm a lot more to hold on to and to use to separate your populations. If you've ever tried clustering on just OPSNI 1 and 2 or just UMAP 1 and 2, you get this kind of weird stained glassy type look where it hard cuts between populations. That you can avoid that basically. If you give it 8 populations or 8 dimensions to kind of separate your data, even if you use only UMAP dimensions, it won't have that kind of stained glass appearance because it has more to hold on to. So you get kind of much more normally distributed populations, buzzing between the edges, and I think probably a more accurate grip on the different populations in your data. So here you can see this is UMAP 1 versus 2. This is the same data set. This is the same population being highlighted. It's all being colored by CD4. So this is CD4 by itself. UMAP 3 versus 4, it's separating out other things. UMAP 5 versus 6, you know, it's starting, the separation is starting to get less clear. But here in this particular CD4 population, it's, you know, scrunched. It's maybe like an 1/8 of the total area here visible. Here it spreads out. You know, there's a lot more difference between the CD4s over here and the CD4s over here. It just so happens that it's all smooshed together with other populations here, but that's what it looks like in 8 dimensions. So number one, give it a shot if you don't mind explaining to your audience what you did and why you did it. Again, you can say Geoff Kraker told me to, and I didn't tell you to, but I said try it, and it's kind of fun. So number one, I did this, and we're going to scroll up here and take a look. So I did an 8-channel UMAP, and then I did FlowSOM on those UMAP markers, and again, these kind of smeary, important surface markers that I use to distinguish populations from each other. And we have that now. Again, you can see those 8 UMAP channels, and I told it to split it apart a bunch. And then I used the 50 meta cluster version. And then I made a figure and some heat maps. And that's what we looked at in the slides. That CD141 positive dendritic cell population ends up in its own cluster with this method. And it didn't end up in its own cluster in the official figure. I don't know if you all caught that, but it is a real population that they gated for manually, even though there weren't that many cells there. But it didn't end up, that wasn't obvious. It wasn't its own cluster in the official kind of UMAP FlowSOM figure. But here, if you go, once it loads, to, let's see, I think it's 25 in this particular 2, 3, 4, no, not 25. Here it is, 32. This is that cluster. So this is just that, only those 141 bright positive dendritic cells, and they ended up in their own cluster. And I didn't like specifically search for them. You know, I checked to make sure that they were in their own cluster, but I didn't do anything above and beyond other than give UMAP more parameters, and then that gave FlowSom more to hold on to when it was separating my heterogeneous population. So there certainly can be dividends. I'm not guaranteeing it'll work on your data. Again, this is just one strategy, but it can help. So consider trying this. If you're not averse to explaining to somebody why they're looking at 8-dimensional UMAP data, just say it's like principal components. People do, you know, 12-dimensional PCAs all the time. You know, it's just like that. Not under the hood. No data scientists in the room. Please, I understand. But that's just something to keep in mind. So that's one kind of fun thing to tackle. The other is that there's other algorithms out there. It's not just UMAP and FlowSom. We saw a great list from FlowJo and from FCS Express. People are adding more of these algorithms into their software platforms all the time, and people are developing new algorithms all the time. So just because UMAP works all right doesn't necessarily mean it's going to be the best option forever. So if it works for you, if it works for your data, that's fantastic. But you can use other things. You can experiment. So in this case, I ran Park on this same data set, which is a clustering algorithm that you don't have input into the number of clusters specifically. It tries to find the right number of clusters, kind of like Phenograph does. So definitely take a look at Park. EmbedSum is a dimensionality reduction algorithm that uses self-organizing maps, kind of like FlowSom does. It doesn't do a fantastic job of clearly separating your different populations, but it is really fast, and it can handle a lot of events. So if you're really kind of pushing the upper limit, you've got like 20 million events, and you're like, man, UMAP definitely doesn't work on this. What can I try to help separate my populations? EmbedSom's a great choice for that. And you can pair it with other stuff. In OMIC, you can take OPSNI, which is a dimensionality reduction algorithm, and you can start OPSNI running by pre-positioning your events. So a lot of the algorithms, like the original TSNI and UMAP and a bunch of them, they just dump all your events into a random pile and then start separating them into their own specific islands. This is kind of like Halloween candy. I use this analogy all the time, so sorry if you've already heard it. But it's like, you know, I think of dimensionality reduction algorithms as like dumping your Halloween candy on the kitchen table after you come back, and you put the Skittles next to the M&Ms, and you put, you know, like the Reese's Pieces next to the M&Ms because they're chocolate and small and round, but they've got peanut butter, so they're in their own little group. And you arrange everything like that. That's starting from scratch, dumping them in randomly. That's what most dimensionality reduction algorithms do. However, if you use other channels, and I'll explain what I mean by this in a second, to initialize these algorithms, basically what you can be doing is like, not start from random, but start from kind of some order that you've already imposed on it. That's like putting all of your chocolate candy in one little baggie and all of the sugar candy in all the other baggie, and then you already have done the lion's share of the work separating stuff. What is Bit-O-Honey? I don't know. Nobody likes those. They can be on the floor. But I'm done with the analogies now, I promise. But basically what you can do is you can have something like EmbedSom or like PCA do some really quick but kind of pre-arranging of your populations. And then you can have OPSNI take that kind of, you know, a little bit of momentum that it adds to it, and then, you know, do the hard work of separating out your Tregs from your other T cells or your 141 positive dendritic cells or whatever. And so in the OPSNI setup page, there's this option down here that's called pre-init embedding X and Y. So if you run, and I'll show you what this kind of looks like. If you run PCA first, and then beneath that PCA, and you now have those PCA channels available to you. Then you run an OPSNI. You can use those PCA channels to start kind of ahead of where you normally would with that random. And this can help, especially for these kind of rare, tough to separate populations. But you can do the same thing with EmbedSOM. So here, I ran EmbedSOM first. It's also really, really fast. Not quite as fast as PCA, but so I started here. This is on 2.2 million events. Started at 136 and it got done at 142. So just like 6.5 minutes. So I ran EmbedSOM first, and then I ran OPSNI and included those EmbedSOM parameters as my pre-initialization embedding. Although you don't have to do it that way, you can use CCR7 and 45RA as these pre-initialization embedding things. And so if you want to make sure that you kind of start ahead to help separate your naive from your central memory from your effector memory, you can do that. It doesn't have to be an algorithm to start from. It can be just important surface markers for you, or 25 and 127, or again, whatever else. It kind of gives you a little bit of a head start. Okay. So wrapping up here, this is the last little conceptual bit. I mentioned this. I said data scientists, are like, well, I wouldn't do that if I were you. But number one, they're not cytometrists, and number two, it's not bad necessarily. And I'll explain kind of what's going on here and just what you have to be cognizant of. Dimensionality reduction algorithms are one way of looking at your data. They're one tool in your toolbox of separating all of your populations from each other. Clustering algorithms are another way of separating all your populations from each other. But regardless of whatever one you're using, you're still going from 30, 40, whatever dimensions down to a lower number of dimensions. And you're representing them at that some way. And so you're making sacrifices, just like globe to a map. It always goes back to that. I'm not done with that analogy. I'm gonna keep saying that. So these tools are just different ways of looking at high dimensional data, and they go about it different ways. So like, what's truth? The truth is, if you could in your head visualize 40-dimensional data, then that would be the truth, because that's actually what that object looks like. It's not a 2-dimensional t-SNE or even an 8-dimensional UMAP. It's a 40-dimensional object, and we make sacrifices when we try to distill that into lower information. So the different ways operate differently, not surprisingly. And so there's basically not 100% concordance between dimensionality reduction and clustering. And so what I'm showing you here is a k nearest neighbors, which is one way to cluster things. It's showing you if you mouse over, and this is not live, I'll explain what this is in just a second. But basically, there's a cell in the middle here. And when you mouse over this cell, it shows you where the 40-dimensional nearest neighbors are to this cell in the UMAP. And you'll notice some of them are actually not right next to this cell in the UMAP. And so that's the point that I want to say is, just because the UMAP gives you an answer doesn't mean it's the absolute truth. It's one imperfect way of looking at a 40-dimensional object. The k nearest neighbors is one imperfect way of looking at a 40-dimensional object. That also is not truth. It's just one way of describing the distance between your events in 40-dimensional space. So if you use dimensionality reduction algorithm parameters to cluster on, you might basically be kind of making assumptions about what's close to each other in many-dimensional space, where it's not actually close to as close as you think it is in many-dimensional space. So can you do this? Should you do this? It depends. I can't tell you the number of times I answer questions that people ask me with, it depends. So I feel bad doing it, but it really is true. A lot of it depends on your panel and what you're looking at and the question you're trying to ask and the answer you expect. So if you want context with your data, I'm happy to talk about it. I can't give you an answer here, but if I take a look at your data, maybe I can add some insight to it. And so this work here comes from Tyler Burns. He's a great kind of cytometry adjacent bioinformatician. And he has a lot of content. In just a second, I have a slide coming up where I'll direct you to kind of where you can find it. But basically, he writes all these fun little scripts and opinion pieces and useful content about cytometry and data science, which is pretty sparse, frankly. So I think it's valuable to take a look at. And so he developed this little package. If you are handy in R, you can go take a look, and you can look at this actual interaction on your real data with this thing called KNN Sleepwalk that he developed. So this is another example of what this looks like. So this is basically one region in dimensionality reduction space, and this is those events, basically what's next to those events in high dimensional k-nearest neighbors space. And most of them are pretty close, but some of them are not. So again, just something to keep in mind. It's not an automatic, this is fine because Geoff said it. It's you're making compromises. But as long as you know you're doing it and you describe it to people and you proceed with that information in mind, I'd say it can be a useful tool. So wrapping up, these are miscellaneous resources. If you have an interest in this stuff, in data science and cytometry combined, awesome, number one. Number two, you might find a lot of useful information at this blog, towardsdatascience.com. And another one, this is a YouTube channel, and it has its own website, statquest.org. They're great resources. They're not cytometry specific. They're broadly data science covering channels, but they do talk about like why UMAP is better than TSNE, and or isn't. Actually, it might not be. There's a whole paper out of it. So they do cool stuff like that. If you're curious about what doing different things, different settings to UMAP does to the results, there's a whole website about how UMAP works and like the guts of it and under the hood, what it's doing and what you should think about with your data. And it's not, again, not cytometry specific, but it is really good insight into how UMAP works. This is that website that I use to pick those colors for that figure. So as I was doing it, basically, there's a lot of data viz tools out there on the internet, but this one specifically allows you to pick two hues and say, I want 8 hues directly matching in between these. If you have 8 little clusters inside of one of your populations, you can copy the hex codes from this website and overwrite the default colors in OMIC. And I'll show you how to do that right now really quick, because man, it just makes the figure so much better than by default. We have like really high contrast colors, which is great, but it is good, I think, to use a whole bunch of better looking colors. Let's see. Select all here, and so now you can see all these colors again. If you want to change what that color is, you can open up this overlay option and click on this, and it gives you a little hex code. And you can paste the hex code from that website into here, and it will automatically bring up the one of those 8 colors that you selected. So it's a great resource to as you're polishing your figures. And then lastly, this is Tyler's blog. Again, he's got a lot of cytometry focused informatics information out there. He posts a bunch of stuff on LinkedIn too. You can check it out. Yeah, he does not work for OMIC, so I'm not like repping one of our own people. I think he's got some insightful stuff to say. And with that, I'll wrap up. So thanks to FlowTex, thanks to SciTech and the team there for making this awesome data set that I have a little bit too much fun analyzing. The cytometry data community. If you haven't gone to any one of their talks or any one of their sessions at Cyto or at these local things, I'd certainly recommend it. They're really cool, innovative people. Thanks to Chris and the rest of the OMIC team for making fun software. So thank you very much.
We convert a lot of imaging cytometry
Mm-hmm.
into
Yep.
physical.
So yes and no. So you can use CSV files in OMIC. So if your image segmentation software will give you nearest neighbor information, if you can record those values, then you can, or any features you want, you know, shape or whatever, you can then use those in OMIC. If you can derive them some way, you can look at them in OMIC. Right now, we don't have a lot of imaging tools just because there's not a lot of people requesting imaging tools. So if you want imaging tools, be a squeaky wheel and let us know. We're happy to prioritize stuff that people care about. Yeah, I know it's a lot. Like I said, we kind of ratcheted it up there at the end, so no worries if you're a little bit like, wow, that was a trip. And if you're like that from the beginning, that's cool too. I know people are just dipping their toes in, so come find me. I'll be at the Dotmatics table, and we can have a chat. Yeah, thanks for hanging out in the last talk of the day. Kudos, extra kudos to everybody.
All right, so this is wrapping up day one of FlowTex, the interactive portion, where we're testing out the practical data analysis and kind of fun. If you guys have an idea for doing something similar or have requests for next year, let us know or join the committee and help us out. We'd love to have you. This is just another way to kind of keep the community involved in this type of learning and training. Other than that, we have 2 more days of FlowTex coming up. So we have tomorrow morning. I believe we're starting at 8:45 Central Time. We can't wait to see you. Tomorrow will be tacos for top breakfast tacos. We're excited about that. First year at FlowTex to have breakfast tacos. You guys online have to. Well, sorry. Maybe we'll post a recipe. And otherwise, if you guys are here in person and need parking validation, we are validating for the directly adjacent UT parking lot. We'll be doing that for the entire conference. Garage, the UT garage. That's on the corner of Pressler and Fannin. Other than that, I believe no other announcements besides have a great evening and thank you so much for turning up. We really appreciate it.